Good morning, everyone. Welcome to the Bio Korea 2022 Bio Live. I am Kim Kyung-mi, and I'll be your MC for this event. Thank you very much for joining us. Bio Live, Day 3. The 3-day long Bio Live that started from on Wednesday in Seoul, Korea, is aimed to promote attendees' technologies and products, and perhaps become a starting point to a newly unexpected partnership with global companies. Embassies and investment institutions from 5 different countries, along with pharmaceutical companies, clusters, local government, both local and foreign buyers, have attended to pursue a new adventures. Today's Bio Live will be heard at the Bio Live Drone as well, where both offline and online presentation will be given. For those who are unfortunately unable to make it here in the studio, they also have recorded videos of their presentations that which will be shown on our official website. And I look forward to your active participation for the Q&A session during the event. Then now let's officially start the last day of Bio Live. The beginning of today's session will be given by the Canadian Embassy. Companies from 11 companies actually from Canada will be introducing their technologies and products for today. And firstly, Jim Pankovic from Bold Therapeutics will present first. And he is here with us in the studio. So, ladies and gentlemen, please welcome him onto the stage.
Thank you very much, and thank you to the organizers and the Canadian Embassy for the opportunity to present Bold Therapeutics to you today. Bold Therapeutics is a Canadian-based, Vancouver, British Columbia-based pharmaceutical company, and we're privileged to present our product to you today. Bold Therapeutics has taken on a major challenge in terms of treating patients that often, once patients are diagnosed with cancer, they're often treated, but these treatments can be ineffective, or nearly all patients will have relapsed or refractory disease at some point during their treatment. Bold has taken on this challenge with their product, Bold 100, which is a revolutionary small molecule that improves patient outcomes, not only by itself as a monotherapy, but in combination with other drugs in various classes, in multiple cancer indications, and in clinical trials has broad utility to improve patient outcomes. So why Bold Therapeutics? Well, we have a versatile technology. We have clinical data from a previous phase 1 monotherapy safety study, and we're currently conducting a phase 1, phase 2 clinical program with some early compelling data. Our phase 2 is ongoing, and we're currently enrolling patients in Canada, the United States, and in Korea. We have orphan drug designations from the US FDA for gastric and pancreatic cancer, and as our clinical data matures, we'll be pursuing breakthrough designation for one or more of our indications. We have an exceptional amount of non-clinical data, which is consistent, and working with our worldwide partners to evaluate the mechanism of action and potential indications for future clinical work. We have an extensive amount of synergy data with combinational therapies and showing that we can work in combination with these therapies. And as a first-in-class product and a unique mechanism of action, we believe we have a large commercial potential to be able to move our products forward through the development program and into commercialization. With that, we've also got a veteran team on our corporate team, considerable amount of experience raising money, negotiating international partnerships, and working as a private company working towards becoming a public company. We also have extensive experience with clinical development, conducting global clinical trials, incredible amount of oncology experience both within the company and through our partners, and successful regulatory interactions with the Health Canada, the US FDA, and the Korean MFDS. Our non-clinical team has extensive experience and has spent much of their time over the last 2 years elucidating the mechanism of action, and we've completed multiple GMP runs for manufacturing for our clinical trials. As you can see, we're also doing a lot of our collaborative work in North America, Europe, and in South Korea. So our international network of researchers that we work with makes a compelling story to be able to partner with Bold Therapeutics. A corporate snapshot, so the company was formed in 2018, and we've been very busy since that time doing clinical manufacturing and getting back into the clinic with Bold 100 in combination with FOLFOX chemotherapy in gastrointestinal cancers. In February, we completed our phase 1 combination dose escalation with Bold 100 and are now transitioning into our phase 2 study, where we're looking at additional patients and looking for phase 2 proof of concept efficacy data in colorectal cancer, pancreatic cancer, gastric cancer, and bile duct cancer. At the same time, we've done an extensive amount of preclinical work with our partners around the world to further understand the mechanism of action and determine how we can move forward and how we utilize that information to move forward in our current indications as well as other oncology indications. Our goal is by the end of this year to be fully enrolled in most of the arms of our current clinical trial and to look at initiating at least 2 additional clinical programs. 2023 will also be a busy year as our data continues to mature from our phase 2 program and as we initiate clinical trials based on this data. Our current clinical trial is really a phase 1, phase 2 clinical program designed to understand in the first part the safety and tolerability of Bold 100 in combination with FOLFOX chemotherapy. We did this and have completed this stage of the study looking at 3 different dose levels of Bold 100 and now are moving into the phase 2 dose expansion portion of the study. This portion of the study will also look at safety and tolerability, but the focus now changes to efficacy and proof of concept data, looking primarily at progression-free survival and secondarily at overall survival and objective response rates in patients that are treated with Bold 100 and FOLFOX chemotherapy. We will be enrolling patients in gastric cancer second line, pancreatic, colorectal, and bile duct cancer. And from some of our early data, we're also looking to explore additional indications in third-line colorectal cancer and esophageal cancer. Some of our early data from our phase 1b portion of our clinical trial, again, keeping in mind that the primary purpose of this study was safety and tolerability. So we were able to show that we can safely combine Bold 100 with FOLFOX chemotherapy with no new grade 3 or grade 4 treatment emergent adverse events. Interestingly, in talking with the physicians treating these patients, one of the comments was that we were able to continue treatment for these patients through an extended number of treatment cycles, with one patient administered out to 15 treatment cycles. Efficacy, while not the primary goal of this portion of the study, we did start to look and evaluate efficacy in this patient population, and from some of the preliminary data, have very compelling and early clinical data showing improvement in progression-free survival over what would be expected with standard of care therapy alone in this very difficult to treat patient population. On the responses, we had one patient that had a 48% reduction in target tumor lesions, a second patient with a minor response with 22% reduction, and a number of other patients that also had minor responses. As a benchmark comparator in terms of the market size potential for a product like this in colorectal cancer, you'll see the data from Cyramza showing that this is a product that is utilized in second-line therapy in combination with standard care, and in 2021 had sales of approximately $1 billion. So there's considerable amount of unmet medical need as well as commercial potential for this indication. An update on where we're at currently with our clinical program in the phase 2 portion. Again, we're enrolling patients at 13 clinical trial sites across the world, 6 in Canada, 2 in the United States, and 5 in South Korea. We have completed the enrollment ahead of schedule in our colorectal cancer arm. We are ahead of schedule in terms of our enrollment in the bile duct cancer arm, and we're trending on target for both our pancreatic and gastric cancer arms for enrollment into our trial. Oops. The mechanism of action, we spent quite a bit of time over the last 12 to 15 months further understanding the mechanism of action and how we might be able to utilize this to further improve our development program for Bold 100. Our Bold 100 is a ruthenium-based molecule and has a multimodal mechanism of action, primarily through 2 mechanisms. On the left-hand side, you'll see that through the unfolded protein response, Bold 100, when administered to patients, prevents the upregulation of the master regulator of the unfolded protein response, GRP78. It's through this prevention of the upregulation that then makes the cells susceptible to apoptosis and cell death. In addition, at the same time, we further understand more about the reaction oxygen species induction. Again, when BOLD 100 is administered to cancer cells, we see an increase in the reaction oxygen species induction, which then results in DNA breaks. Again, these DNA breaks lead to cell cycle arrest and ultimately to cell death and through apoptosis. Again, both of these mechanisms likely work in all cancer types, but different cancer types likely have a different contribution of each one of these arms to the overall effectiveness. GRP78 is important in that increased levels of GRP78 have a poor prognosis for patients that have this elevation. So this is one of the key targets for our clinical program. Interestingly, here's a little bit of the data that we've been able to gather over the last year or so, really exemplifying some of the usefulness in vitro and in vivo to be able to understand how the product works. On the left-hand side is an experiment that we've done with the Moffitt Cancer Center in Tampa, Florida. This is where our clinical investigators were getting biopsies from pancreatic patients, extracting those biopsies, and then submitting them to various chemotherapies in this live tissue sensitivity assay. In this assay, on the top panel, there's 5 patients that are being exposed to either a control arm, BOLD 100 alone, gemcitabine paclitaxel, or the combination of gemcitabine paclitaxel and BOLD 100. Similarly, on the bottom, it's a same experimental design, the difference being the chemotherapy that's being administered, with it being FOLFIRINOX. In all cases, BOLD 100 has a monotherapy effect, and interestingly, the chemotherapies also have an effect, but surprisingly, with the green bars is where the most interesting data is. When we combine the standard of care chemotherapy with BOLD 100, we see the most dramatic effect in terms of inhibiting the tumor cell viability. On the right-hand side is some data we're doing with Queen's University in Belfast, understanding, overcoming some of the resistance to proteasome inhibition in multiple myeloma. The researchers at Queen's University in Belfast have developed a proteasome-resistant cell lines to either the proteasome bortezomib or carfilzomib. And interestingly, when combined with these in a sensitive cell line, BOLD 100 has an additive effect. But importantly, is in these resistant cell lines that have been developed, when BOLD 100 is added back to either bortezomib or carfilzomib in combination with the resistant molecule, both of them have shown strong synergy and the ability to overcome the resistance that is inherent in those cell lines. We've also been able to demonstrate over a wide range of different anti-cancer therapeutics. On the left-hand side of this panel, you'll see a number of cell lines, different indications, as well as different chemotherapies and targeted therapies. And in nearly every case, you see it when combined together with BOLD 100, you'll see either a synergy or an additive effect. On the right-hand side is an animal model in a very difficult to treat cisplatin-sensitive gastric cancer model. In this study, we actually dosed the animals with a subtherapeutic dose of BOLD 100. So you'll see the vehicle in BOLD 100 having a similar effect. Cisplatin is the standard of care in gastric cancer, and you'll see that there's a minor response when administered on the orange line. But interestingly, again, when we combine cisplatin plus BOLD 100 in the green line, you'll see the most dramatic effect in keeping the tumor reduction or tumor burden down. We have strong and robust worldwide intellectual property coverage for both composition and matter of our drug substance and drug product, as well as method of manufacturing. And this includes in all the major markets, including the US, Europe, Canada, and beyond. We do have a partnership with a local pharmaceutical company in Korea, Hanapharm. This was an option agreement that was signed back in mid-2020 and includes both milestone payments and development collaboration as we move forward. We are in the process of raising capital. We're getting ready to close our Series 1A round of capital, raising $5 million. That will give us about an 18-month runway and allow us to be able to complete our current phase 2 clinical program and get some of our proof of concept data later this year. Once we actually get some of that data, we'll move into a Series B round, raising additional resources to conduct additional clinical trials and to be able to move our product development process forward. So some of the milestones that we've completed or will be completing over this next year, and 2021 was a busy year for Bold Therapeutics, and definitely 2022 will continue to be busy as well. We completed our phase 1b clinical data in our advanced cancers. This again was primarily safety and tolerability data. In March, we also then initiated our phase 2 portion of our trial, enrolling patients at 13 clinical trial sites worldwide. In April, we presented some of our non-clinical data at the American Association of Cancer Research. In June, we'll be presenting some of our clinical data from our initial phase 1 portion of our current combination trial at ASCO. We will be shortly closing our Series 1A round, and we're expecting in the fourth quarter of this year some interim phase 2 data from at least 2 arms of our current clinical program. This will be primarily some of the efficacy, proof of concept efficacy data with progression-free survival. And as we move into next year, we're anticipating starting some additional clinical programs, closing a second licensing deal near the end of the year, and then initiating our institutional round of fundraising. In 2023, we'll be getting additional efficacy data from our current clinical program, including additional progression-free survival and overall survival data, and then looking at the potential IPO in the second half of 2023. So I'd like to thank you for your attention today, and if there's any questions, I'm available here, or I will be at the Canadian Embassy booth that is in the exhibit hall later this afternoon. Thank you very much.
Thank you very much. So do you have any question on site from the floor? If you have any question, please raise your hand and we'll get you a microphone. Okay, then with that, thank you very much, Executive Vice President, for that excellent presentation. Also, you can send a question to a presenter online. Thank you. Okay, moving on to the next, I would like to introduce the next presenter, Ms. Trina Racine, the Associate Director, Vaccine Development at the Vaccine and Infectious Disease Organization. So she is here with us in the studio. Without further ado, the floor is now yours. Please welcome her.
Good morning, everyone. I'd like to start by thanking the organizers of BioKorea as well as the Canadian Embassy for inviting me here to present to you today. As mentioned, my name is Trina Racine. I am the Associate Director of Vaccine Development at the Vaccine and Infectious Disease Organization, also known as VIDO. VIDO is a research center within the University of Saskatchewan, and we operate Canada's largest high-containment infrastructure. VIDO has been in operations for over 45 years, and our more than 160 personnel focus their research on pathogens that infect both humans and animals. What is circled here is our location on the University of Saskatchewan campus. So of the 160 personnel currently at VIDO, more than 45 of them are PhD level scientists that have come to us from more than 25 countries around the world. VIDO is ISO 9001 certified, and as I mentioned, we operate more than 225 million Canadian dollars worth of high containment infrastructure. Over our history, VIDO has commercialized 10 different veterinary vaccines. 6 of those were world firsts. This is a picture of our International Vaccine Center, or InterVac building. This is our high containment level 3 or biosafety level 3 agriculture facility. The glass building here, it contains our level 3 laboratory space, and the gray building in the background is where the animal labs are located. The InterVac building can house a wide range of species, everything from small rodents, mice, hamsters, as well as ferrets, and then we also can work with bats, and all the way up to much larger animals, such as alpacas, and what we have pictured here are bison. So VIDO is focused on a One Health approach to our research, where we, as I've said, do conduct research on both pathogens that infect humans and animals. We conduct research looking at the mechanisms of infection as well as transmission and the resulting pathology. We also do a lot of animal model development for infections. And our goal at VIDO is to develop novel vaccines and therapeutics that we can eventually license to external organizations to bring them to commercialization. our ability to bring novel products towards that commercialization goal is going to be aided by our new Vaccine Development Center, which is a pilot-scale GMP manufacturing facility. In addition to conducting our own independent research at Vito, we also operate as a contract research organization. We work with external companies and clients from around the world to test their novel vaccines and therapeutics in our animal models of infection. In the last 2 years alone, we've worked with more than 100 organizations from around the world on COVID-19 projects. And being a center at the University of Saskatchewan, we also have a big focus on knowledge sharing and training. And we do this by presenting at conferences such as these, as well as publishing and doing media interviews. Our focus on training the next generation of researchers as well as GMP manufacturers is evidenced by the more than 50 graduate students and postdocs that we have at Vito. As I mentioned, we have a One Health approach to our research, and we do this by studying pathogens that affect both humans and animals. On the human side, we look at a variety of pathogens, some of which are listed here, including SARS-CoV-2, Zika, MERS, and whooping cough. On the animal side, we focus on pathogens that affect producer animals, such as cattle, swine, and poultry. And it's because these pathogens can cause severe and significant economic loss to farmers in Canada. And we also look at a number of pathogens that affect wild animals, such as the prions that cause chronic wasting disease. So in the last 5 years, Vito has developed and licensed 2 new vaccines, and we've also brought forward a vaccine for COVID-19 into the clinic. We've received 5 research and development awards, and we've had the opportunity to work with more than 250 different companies and organizations, academic researchers, where we have been able to provide our expertise and access to our infrastructure. We've made over 1,000 publications and presentations and developed 11 different animal models, some of which are demonstrated here, for example, bovine TB, COVID-19 in the ferrets, as well as a MERS, a Middle East Respiratory Syndrome coronavirus model in alpacas. So Vito has been one of the leaders in Canada as responding to the COVID-19 pandemic. And we have done this by being the first organization in Canada to isolate the SARS-CoV-2 virus from a patient sample. An individual returned from China and was infected, and we were able to isolate the virus from that sample. We're also the first organization in Canada to produce an animal model of infection. That was the ferret model. And then we also developed the hamster model of infection. We were the first organization in Canada to have a COVID-19 vaccine in human clinical testing. And we have been a part of 3 different World Health Organization expert groups. And as I mentioned previously, we've also engaged with over 100 companies to test their novel vaccines and therapeutics in our COVID-19 models of infection. And one of those companies has now had their product licensed for public use, and 6 others are in clinical trials. And that just demonstrates that the quality of data that we can generate in our preclinical research environment, that it is acceptable for use from the regulatory agencies. Now, what I've just been discussing is what we at Vito are currently doing, but we are also expanding our vision and goals for our organization. And that vision is now to become Canada's Centre for Pandemic Research. And what this will mean is that we will be Canada's leading institute for emerging infectious disease research. And we will be able to do everything from early initial discovery research at the bench, all the way up to preclinical testing in our high containment infrastructure, all the way up to manufacturing GMP material for use in human clinical trials. And we can do this all in-house, all in one location. And what that really means is that we will then be able to not only save time, because everything is located in one place, but also save a significant costs, because we won't have to go to a number of different locations for preclinical testing, for manufacturing, et cetera. And as we become Canada's Centre for Pandemic Research, we will be increasing the number of trainees and highly qualified personnel that we have working at Vito. We have received significant support from all levels of the Canadian government for this vision from the federal government, from our provincial government in Saskatchewan, as well as from our local municipal government in the city of Saskatoon, where we are located. And this support for our vision has come in the way of significant investments, more than 140 million Canadian dollars in funding over the last 2 years to become Canada Centre for Pandemic Research. The majority of this funding has been to improve our infrastructure, but some of it has also been to support the clinical development of our COVID-19 vaccine, for which we are in phase 2 clinical trials, as well as in a phase 1/2 trial for our booster indication, and for which we are currently looking for a licensing partner. Some of that funding has also come in the form of research grants to our scientists. And so, like as I mentioned, the majority of this funding has gone towards infrastructure and is going towards infrastructure. And now I'm just going to point out a few things of how we're building to become Canada Centre for Pandemic Research. And so the first is the building of a new containment level 2 animal facility here. This is the original Vito building in the middle. On this side here, we have a current containment level 2 laboratory space, a building that was built about 20 years ago. And this new facility here is going to expand the number of species and the types of species that we can work with. We will be able to conduct research with insects and ticks, all the way up to non-human primates. We are also upgrading about 10% of our containment level 3 agriculture facility into the containment level 4, so the highest level of containment that exists. And we're doing this so that we can then be able to respond to any emerging pathogen, any pathogen that currently exists or one that may emerge in the years to come. And another significant investment in infrastructure that we're doing is the development or the construction of our vaccine development center, that GMP manufacturing facility that I mentioned earlier. And so that manufacturing facility is going to be located in the top 2 floors of the Intervac building here in that glass structure. And what this does is it allows us to have a GMP manufacturing facility that is level 3 capable. So we'd be able to produce inactivated or attenuated level 3 vaccines. And so our manufacturing facility is going to be very versatile in that we'll be able to produce vaccines for both humans and animals. In North America, we'd be able to bring those veterinary vaccines all the way to licensure. And we'd also be able to produce up to phase 2 clinical trial material in our facility. We'll be able to manufacture live attenuated, inactivated, viral vectored, recombinant protein vaccines, as well as DNA and RNA-based vaccines. And so we'll have a wide variety of options available, and this facility will be open to our own internal researchers, but also to external clients who may want to have their products manufactured in our facility. And our niche is going to be focusing on process development, so that step between taking a vaccine from the bench and bringing it into clinical trials. There's a lot of work that needs to happen in that arena, and so our staff is very well versed in that, have over 50 years of combined experience in manufacturing, and we believe that we would be a very good big asset for small biotech and academic researchers who are looking to enter that clinical space. And so with that, that is how we are becoming Canada Centre for Pandemic Research, all the way from discovery to the ability to produce clinical trial material. And so with that, I'd just like to thank everyone for their time, and I'd be very happy to take any questions. Thank you.
Thank you very much. So, do you have any question from the floor? That was excellent presentation. It was good to know about the research center. Any questions or comments? Okay, so thank you very much once again. So once again, if you have any question or comment later on, you can send a question to the presenter. So you can visit our official website, or you can scan the QR code. Thank you very much. And from now on, ladies and gentlemen, we'll watch the pre-recorded videos from 9 Canadian companies. The order of the presentation will go as Alert Life Science, Bioxilium Research, Blue Dot, Cyclica, My Intelligent Machines, Neuradox Incorporated, Ovenza, UHN Research, and finally, Variational AI. So with that, please enjoy the video.
Hello, my name is Harold Stover. I'm the CEO of Alata Life Sciences and very happy to be here to tell you a bit about our technology to enable cell-based therapies for type 1 diabetes and other indications. I will tell you a bit about our cell delivery technology shown on this slide on the left and how we are using it to enable allogeneic cell therapies. Our lead indication is type 1 diabetes. I will show you some technical and preclinical validation data, talk about near-term value drivers, and other future therapeutic indications. Our board of directors includes myself as well as Maria Antonakos, my co-founder and COO, as well as Ian Delaney, Mark Kremble, and Julia Daniel, who represents McMaster. All three were early equity investors in Alata. The potential of cell-based protein replacement therapy, which is our version of cell-based therapy, combines a number of indications where a single protein is missing in a patient. In the case of type 1 diabetics, this would be insulin. In hemophilia, this could be a blood clotting factor. In addition, there are other disorders where a more complex mix of proteins needs to be produced by transplanted therapeutic cells. All of these therapies have in common that often stem cell-derived therapeutic cells need to be implanted into patients to function for a significant period of time, producing their peptide-based payloads. The promise of allogeneic cell therapy is huge, but we are still encountering challenges that have to do with the need for immune protection of some of these cells, the need for metabolic connection, especially in the case of islet of Langerhans, and the need for enhanced persistence in the case of mesenchymal stem cells. At Alata, we are addressing these challenges through designing molecular approaches to shield transplanted cells from different immune attacks. We have developed over many years in my academic lab at McMaster a set of immunoevasive materials that we are using not just to encapsulate cells, but actually to provide a supportive housing that maintains their cell function, allows very efficient metabolic exchange, secretion of therapeutic agents, while keeping the immune system at bay. This technology is now in-licensed into our startup company Alata, protected through a number of issued patents, and we've developed this into a safe, sustained, localized, robust, and scalable technology to enable cell-based therapies. An example of a particular early shape factor is shown on the left. This is a roughly 1 millimeter in diameter hydrogel bead that we implanted into the peritoneal cavity of an immunocompetent rodent. And after 6 weeks, when we took it out, we saw very little foreign body response to this implanted biomaterial. And that suggests that any therapeutic cell payload inside such capsules or other shapes of hydrogels would be well protected from immune recognition while being able to function therapeutically. There are many indications for such a platform technology. As I mentioned, we will focus on donor islet transplantation into type 1 diabetics, in particular highly brittle type 1 diabetics. That will be our validation first indication. Subsequently, we plan to expand to stem cell-derived beta cells, as well as lysosomal storage disorders, neurodegenerative diseases, cardiac and inflammatory indications, as well as rare blood disorders. There have been a number of remarkable deals in this space. Many of you will be familiar with the acquisition by Vertex of Semma Therapeutics, which included the therapeutic cells as well as certain delivery technology. At the same time, or about the same time, was also a partnering of Eli Lilly with Sigilon for encapsulation technology. Our first indication will be transplantation of human donor islets into type 1 diabetics. This is a large global need, which is currently serviced in part by transplantation of human donor islets with immune suppression. We propose that use of our immune-privileged hydrogels will expand the eligible patient pool beyond the most brittle patients and reduce the cost and risks to the receiving patients because we can remove the need for systemic lifelong immune suppression. At a later stage, we expect to be able to transplant stem cell-derived beta cells into general type 1 diabetes patients as well. This slide shows some in vitro de-risking data. We are using cell attachment assays to measure and de-risk capsules against immune recognition. The panel in the middle shows capsules that were explanted from immunocompetent rodents after 2 to 6 weeks of transplantation. And we also recently were able to retrieve blank capsules without fibrotic overgrowth from pigs, and this is a significant advance. This slide shows that encapsulated as well as free islets respond very efficiently to glucose stimulation. We also tested the autodiffusion of FITC labeled insulin and find a proxy for very rapid metabolic connection. Finally, our capsules can contain rapidly reproducing beta cell lines, an additional safety feature for FDA and other regulatory institutions. In terms of animal studies, we have used immunocompetent C57 black 6 mice, STZ diabetic, that we have transplanted with encapsulated rat islets as well as encapsulated human donor islets. And we have seen very rapid control of these animals' blood sugar levels to normal levels. We have done this in many parallel trials with many different timelines, including for human islets, where of course there's a bit of a mismatch between the human insulin and rodent needs. Glucose tolerance tests also show that in red, transplanted islets are able to correct the blood sugar of otherwise diabetic mice as well as happens in normal mice. So we show here a set of data that involves rat islets transplanted into the same immunocompetent mice for as long as 340 days. And that puts us in a very good position vis-a-vis a number of other players in this field, as we are using marginal islet loads of about 1,000 islets per mouse. In future, there will be stem cell-derived beta cell payloads. We are preparing for this by showing that we can encapsulate both individual cells as well as stem cell-derived clusters. These maintain their activity. We can show that we can grow clusters within capsules, and where they are loaded with suicide genes, we can trigger ablation of these cells. That's again a safety feature that's well received at the FDA. Our path to the clinic in terms of type 1 diabetes then involves transplantation of human donor-derived islets into brittle type 1 diabetics. We are currently preparing by discussion with clinical trial partners. We have retained a CRO to develop our plans on toxicology and clinical trial design, as well as GMP synthesis of our hydrogel formers. So we are looking forward to an FDA pre-meeting either later this year or early next year in preparation for an IND submission in 2023, early 2024. Our first clinical trial will involve 10 to 12 patients. It'll be a single arm, no controls, because the pathogenicity of diabetes is well understood. We have very good discussions with leading clinical trial centers in North America that are very receptive to working with us because we can help them expand their patient pool. We have a number of technologies in our pipeline. The examples I showed relate to technologies that are at the early IND enabling stage in small as well as large animals. We have additional technologies that are further de-risked and optimized for different indications. That concludes my summary of our technology. Thank you very much for participating, and we would welcome any questions. You will see where you can reach us. Thank you very much.
Hello everyone, my name is Jamie Padros and I'm president of Bioxilium Research. Before starting my presentation, I would like to thank the Trade Commissioner Service at the Embassy of Canada in Seoul for organizing this virtual trade mission to BioKorea 2022 and for selecting BioAuxilium to participate on the Canadian business delegation. I will start this short presentation by giving you some background about BioAuxilium Research, who we are and what we do. Then I will briefly describe our enhanced proprietary TR-FRET technology, along with our competitive advantages, comparative data to other TR-FRET technologies, and our portfolio of products and services offered to the life science research community. I will then summarize the key take-home messages. BioAuxilium Research is a biotechnology company founded in 2013 by former PerkinElmer scientists. We are located in Montreal, Canada, at the Admare Innovation Center, which is a state-of-the-art and open-access drug discovery hub. Our focus is to provide state-of-the-art tools based on our enhanced proprietary TR-FRET technology, Thunder. Over the last years, we have licensed hundreds of cellular TR-FRET assay kits to a global reagent manufacturer. In 2019, we pivoted our business model to develop and manufacture our own line of TR-FRET cell signaling assay kits based on the Thunder technology. The first panel of Thunder kits was launched in 2020 and additional products were launched this year. Our goal is to accelerate biomedical research by providing top-quality, fully validated yet affordable TR-FRET assays that generate reproducible and consistent data. We also provide a range of expert customized services in assay development. Our unique selling point is certainly our expertise and experience. We have a track record of developing hundreds of commercially successful assay kits for drug discovery and basic research. Let's now review what's TR-FRET and how our technology works. The Thunder technology combines FRET with time-resolved measurement of fluorescence. Foster resonance energy transfer, or FRET, is based on the transfer of energy, not fluorescence, between two compatible fluorophores, a donor and an acceptor, when in close proximity. Thunder uses an europium chelate as the donor fluorophore and a small far-red fluorophore as the acceptor. In a typical sandwich immunoassay application, one antibody is labeled with the europium chelate and the second antibody with the far-red fluorophore. When the antibodies bind to the analyte, donor and acceptor fluorophores are brought close together. Excitation of the donor europium with a flash lamp or a laser triggers a transfer of excitation energy to the acceptor, which in turn emits a specific TR-FRET signal at 665 nanometers. The intensity of this fluorescence is proportional to the level of analyte in the sample. Since the transfer of energy occurs only when the donor and acceptor are in close enough proximity, FRET-based assays are homogeneous. That is, they do not require any washing or separation steps to remove distant donor and acceptor partners. Another key advantage of TR-FRET is that data can be expressed and analyzed as the ratio between the fluorescence of the acceptor and the fluorescence of the donor. The use of the ratiometric measurement further increases data reproducibility because it normalizes small pipetting errors and also corrects for some compound artifacts. The key advantage of using a europium chelate as the donor fluorophore is that it emits long-lived fluorescence that lasts for milliseconds, so the specific FRET signal can be measured in a time-resolved manner. How the time-resolved measurement works is shown in this figure. With a TR-FRET compatible plate reader, excitation of the europium chelate by a pulsed light source is followed by the delay and then the signal measurement. This allows short-lived fluorescence to decay before a measurement is made. This eliminates all fluorescent background caused by the sample and plastic microplate and also by direct excitation of the acceptor, which increases assay performance. Overall, because of their excellent temporal and spectral resolution, the Thunder TR-FRET assays exhibit very low background and high signal-to-background ratios, resulting in higher sensitivity. The tear threat detection mode brings additional benefits. Thunder assays have a robust add, incubate, read format that doesn't require any washing. The high resistance of the europium chelate to photobleaching and very long signal stability allows the reading of assays as many times as needed. Thunder assays are easy to automate and miniaturize for screening applications and are also cost-effective. Thunder sets a new benchmark for tear threat assay kits for three reasons. First, these kits were designed with quality and performance in mind. They use a pair of donor-acceptor fluorophores showing exceptional spectral compatibility and tear threat signal. We conduct extensive testing of labeled antibody pairs using optimized lysis buffers for the highest tear threat signal. And we also conduct extensive assay optimization to ensure specificity, reproducibility, and optimal assay performance. In addition, these kits are fully validated. They undergo a stringent validation process using lysets from cells treated with pathway-specific modulators to further confirm target specificity and demonstrate performance in real assay conditions. We include the validation data in the technical data sheets, the most complete in the market. Lastly, we manufacture all kits in-house and we verify lot-to-lot consistency using a functional QC assay where each new lot is compared to the previous lot. In short, Thunder is a cost-effective assay platform that provides easy access to the tear threat technology. As part of our product validation efforts, we have benchmarked some of our key assay kits against two competitive tear threat platforms. Here, we show comparative data for FOFO-AKT-PAN phosphorylated at serine 473. Specifically, the three different assays were compared side by side for their capacity to detect FOFO-AKT-PAN stimulation upon treatment of MCF7 cells with insulin. Following treatment, cells were lysed and the phosphoprotein measured in the lysate using the corresponding reagents included in each kit. The summarized data clearly show that while the three tear threat assays exhibit comparable pharmacology, the Thunder assay shows the highest signal-to-background ratio. We have a focused and expanding portfolio of products that include intracellular cell signaling assay kits for detection and quantification of phosphorylated or total endogenous proteins in cell lysates, biomarker assay kits for detection and quantification of key human cytokines in cell supernatant, a cell-based kit for detection and quantification of cyclic AMP, and generic assay reagents conjugated to either our europium chelate or the far-red acceptor dye for assay development. We offer a range of custom services that includes assay development, including the conversion of ELISA to a no-wash tear threat assay kit development, which is based on our industry-validated new product development process, and antibody conjugation to our pair of europium chelate and acceptor dye, or biotin. Our competitive advantages are our more than 30 years experience developing robust biochemical and cell-based assays amenable to high throughput screening, and our standing prices and shortest turnaround time on the market. In summary, Bioxyrium offers world-class assay kits and services at an outstanding value to biomedical researchers. Our products are based on our enhanced proprietary tear threat technology platform. Our value proposition is simple. We provide high-quality, highly validated assays supported by comprehensive and transparent data sheets, fast delivery, and exceptional technical customer support. In addition, our products are affordable. We propose a focused and expanding portfolio of reliable assay kits and reagents designed for accelerating the pace of drug discovery and basic research. Overall, the Thunder assays provide a compelling alternative to ELISA and other expensive tear threat technologies. Lastly, we offer a wide range of custom services. If you have questions, please contact us or our local distributor 2NC Bio. Thank you very much for your attention.
Hello everyone, my name is Dev Karan Singh and I lead the growth and strategy here at Blue Dot. As an organization, we equip our users with epidemic intelligence data and subject matter expertise so that they can start taking the right data-driven critical decisions with more confidence and certainty. And it all started with our founder and CEO, Dr. Cameron Khan, who is also an infectious diseases physician and has been laying the academic foundation of our platform for over past 2 decades. We formally took shape of an operating organization back in 2013, and we have been servicing our clients and users ever since. Our artificial intelligence platform was one of the first globally to capture the emerging risk of COVID-19 and release the associated notifications and risk assessments to all our users. And this happened on the morning of 31st December 2019 itself, almost 7 to 8 days before any official public health announcement came out in this regard. This, of course, helped our organization gain a lot of media attention, including our Honorable Prime Minister Justin Trudeau speaking about the Blue Dot capabilities and strength of our platform in an official press conference as well. Bringing me to the type of organizations that we work with, when it comes to the government, we work with ministries of health, public health organizations, defense and national security organizations, and research institutes that are supporting different government entities in their decision-making workflows. One of them being Korea Research Institute of Bioscience and Biotechnology that we have been working with successfully for almost a year now. And when it comes to the private sector, we work across different industry verticals, be it the travel industry, that is major airlines, airports, pharmaceutical, therapeutics, or PPE manufacturers as well, hospital chains, fast-moving consumer goods, oil and natural gas industry, to name a few. And to help you understand what is the science behind the infectious disease intelligence that we provide, I have put together this small diagram to help you understand the different inputs and outputs of our platform. Starting with the inputs, our platform scans over 300,000 articles of information captured from over 30,000 sources of information on a daily basis in over 65 different languages. And that includes, of course, the scientific data, travel data, any kind of global mass media, or anonymous mobile devices data when it comes to people mobility as well, domestically or globally. And the outputs of our platform is the necessary data, mathematical and scientific data and information around infectious diseases surveillance, both domestically and globally, and their associated threat assessments, that what kind of impact can they have on your organization, on the economy, on the health of the people or populations, around the business continuity, and demand and supply of products as well. Right? And all this data is made available in the form of raw data or APIs that help connect our data seamlessly with any kind of data visualization or decision-making tool that you're using internally. If any support is required to equip your organization with the right technology or subject matter expertise to build new models using our data, that is also available in the form of enablement services. Talking a bit more about the support ecosystem that we have, especially when it comes to South Korea and our experience of working in this wonderful country, we have three important pillars, support pillars that we have in place to extend maximum support to our end users. The first one being the Canadian Embassy itself, who helps us understand and also our end users understand the trade expertise, any kind of regulatory activities which are required for Blue Dot and the end users to seamlessly work with each other. At the same time, we have a strong on-ground support, both pre and post-sales, technical and non-technical, which is extended through our channel partners, that is Korea Industrial Information Service, who has been working across industry verticals in South Korea for over 2 decades. At the same time, we have a very strong customer success team as well, who work remotely with the end users and extend the right subject matter expertise for their technical and non-technical questions to help them easily adopt and keep using our platform in short, medium, or long term, depending upon the needs of our end users. Bringing me to what lies ahead in the future. So as I mentioned earlier, we equip the public sector, especially with the augmentation of infectious disease surveillance and the associated risk assessments, that is what impact these infectious disease activities happening globally or locally can have, say, on the public health purview overall, helping them take the right decisions around reducing pressure on healthcare infrastructure, designing the right travel or public health policies, taking the right decisions to ensure there is minimum disruption to the economy and the overall public health ecosystem in any particular geography. And when it comes to the private sector, helping organizations take the right decisions around their R&D development acceleration, anticipating changes in demands of their products, building a stronger supply chain resilience, and also ensuring the human resources and operations both domestically and globally. With that, I come to an end of my presentation and would be always open for anyone who is interested to learn more about our technology, about our products and services. Please feel free to reach out either directly to me or our support ecosystem, that is the Canadian Embassy, or our channel partners, that is Korea Industrial Information Service. Thank you so much.
Hello, and firstly, thank you for the opportunity to present to you the work that we're carrying out at Cyclica, which is a company based in Canada and really focused on accelerating the discovery of new chemical molecules. The company was founded in 2013 and since then has been focused really on the development of technologies and tools to help us understand the complex pharmacology of a molecule when it enters the human body. We all know that when a molecule enters the human body, it has both an on-target effect and also an off-target effect. And the idea that Cyclica has had and has since evolved over the last 8 or 9 years is to develop technologies in machine learning and deep learning that help scientists address both the on-target and the off-target effects of their molecules. To do that, Cyclica has built an AI-enabled polypharmacology platform which is providing world-class multi-objective drug design for both high and low data targets. Our platforms utilize a proteome-wide lens to evaluate how on and off targets occur between molecules and potential proteins that may be susceptible to their interaction. Our engines, Matchmaker and Poem, are a deep learning engine and a machine learning engine which marry the structure-based approaches that generate high quality predictions at a fraction of the time, really compared to other techniques. For Matchmaker, we are able to encapsulate a proteome-wide evaluation of pharmacology in real time, and as the name suggests, it makes a match between the protein of interest and the molecule in terms of the design that we may be carrying out, that enables us then to improve the molecular design and enable us to help identify potential molecules that could interact with the proteins of interest, if that is your target, but also in the design of those molecules, apply evolutionary pressures to include also ADMET and other physicochemical properties, as well as potential anti-targets that you may wish the molecule to be designed away from. All of this can happen at a fraction of a second, and our protein-wide data system, the deep learning engine that we have built, means that we can address protein targets across a wide range of therapeutic areas. To enhance that even further, we have now integrated AlphaFold 2 into Matchmaker, thus creating a database which now has approximately 200,000 protein pockets that can be targeted from approximately 18,000 human proteins, which have been structurally enabled in one way or another. And with the technology that we have at Cyclica, we do not need X-ray crystallography data to enable us to design chemistry to enhance the chemical programs of companies, but we can actually work with less precise structural information such as homology models, cryo-EM models, where we can actually then utilize those in our matchmaker system to identify compounds of interest. The breadth of the proteins that we can now address means that we can address both human and other species rapidly, and it allows us to help address problems across, again, wide therapeutic areas, and also address a broad range of drug discovery programs at the same time, due to the power of the system that we have and the way in which the model is utilized to design chemistry. Our success with our partners through the interactions that we've had and from the data that we have received back from our partners gives us an indication that we're approximately 60 to 65% successful in generating what we call actionable hits. These are chemicals which provide activity against a protein target of interest to the partner, and these data have been generated after the partner has synthesized the chemistry that we've designed and then carried out the biological assay to test the molecules that we've provided against the biological protein of interest. And these data coming back to us give us approximately a 60 to 65% success rate in the design of chemistry. That is across a broad range of target classes and also has led to success in several diverse therapeutic areas, some of which are highlighted here. So, in addition to having built the deep learning and machine learning engines that we have within Cyclica, which have helped us address the problems that partners come to us with in different therapeutic areas and within different target classes, we have also been utilizing the engine to establish a program of portfolio compounds for Cyclica's own pipeline. And over the last 2 to 3 years, we have designed and built a pipeline of approximately 50 or so programs, which are highlighted here. And since our Series B, when we've been developing further the technology, we now have up to 50 programs established across the different stages of drug discovery. And as you can see, it also reflects the breadth of the platform can address in terms of the different therapeutic areas that we have programs evaluating it. So for Cyclica, the idea of basing our technology since 2013 on addressing the needs of a polypharmacology of a drug once it enters the human body, the design of the scientific and technical platforms using deep learning and machine learning to address those needs of understanding the polypharmacology of a potential molecule has enabled us not only to help companies de-risk their programs and identify potential off-target interactions, but also helped us to design chemistry that has enabled activity against targets of interest across a broad range of protein classes and therapeutic areas for both the partners and for our own established pipeline. If you have further questions or you wish further information, then please do come and send me an email to verndibiase@cyclicarx.com and be happy to explain further detail how we designed the chemistry and how we partner as a company. Thank you very much for your attention.
My name is Sarah Jena, CEO and co-founder of My Intelligent Machines, and that's a pleasure today to present you our company. Our company is a software company. We're developing augmented intelligence software that enable life scientists working in biopharma industry to leverage systems biology and advanced AI to maximize drug efficacy. A little bit of history on MIMS. We're based in Montreal. We have to date 30 employees, and we have a validated technology that is currently used by large pharma, but also smaller organization and hospital, research institute of hospitals. We start to be renowned internationally for our expertise in multi-omic solutions. And we got also different prestigious prizes over the past years, such as Top 100 Tech Company in North America, but the Red Herring. The issue that we're addressing is the last big problem of biopharma industry, which is drug efficacy issues, which is associated to the fact that patients with the same manifestation of a disease will respond differently to a given drug. That leads to 70% failures of phase 2 and 3 clinical trials and nearly $20 billion loss per year to the pharma industry. And the way we resolve it is by providing life scientists working in biopharma company with a software as a service, so a software in the cloud, allowing them to leverage systems biology, distributed computing, and machine learning to help them better understand patient heterogeneities, to better understand why patients with the same manifestation of a disease will respond differently to a treatment, to identify better drug targets, knowing to which patient this drug target is appropriate, and to identify good responders at preclinical stage, so to help them prioritize indication at preclinical stage, and also accelerate companion test development. We see our software as a life scientist best ally throughout drug development. Our clients currently use our software from very early into the drug discovery process into clinical trial. So very early at the discovery stage, they use our software to classify patients' population based on their multi-omic or mono-omic data and get molecular and clinical characteristics of each subgroup of patients. And they identify targets for each subgroup of patients or identify targets that are appropriate throughout the group, knowing their differences. At preclinical stage, they will use again our solutions once they have an asset, a new drug that is developed, to identify what are the indications with high expected response rate and to prioritize the indication that they will use for their first clinical trial in order to increase the success rate of the clinical trial. They will also use our biomarker discovery tool to add and to identify first a first set of genes that they might add into their nanostring panels. And during their clinical trial, they will continue to use our biomarker discovery to refine these biomarker lists and signatures to accelerate companion test development. The fact that all this is done from discovery to clinical into a single platform enables and accelerates reverse translational research into smaller and larger organizations. So MIMS is a university spin-off. We're capitalizing on more than 50 years of cumulative experience in systems biology, machine learning, AI, and more than 35 peer-reviewed publications. I myself, a former Canada research chair in systems biology, have been working in this field for 20 years, and I've been developing an expertise in integrating multi-omic data to infer genetic interactions, and you will understand a little bit later in this presentation how we are using genetic interaction networks. My two co-founders, so Abdullah Baniri Diallo, is a renowned bioinformatician. He received in 2018 the Next Einstein Fellow Prize for his career at the interface of bioinformatics and machine learning, and has also a deep expertise in natural language processing. And Michael Camus is an AI specialist who worked actually develop different technologies, autonomous systems that he applied in robotics project, for example, with the European Space Agency on the Galileo project. And over the past 10 years, we transposed his technology to the life science space. And his technology is used at the heart of the first part of our product, which is an intelligent cloud platform. enabling distributed computing. So the fact that our platform enables distributed computing actually enables our users, the users of the platform, to select within a minute a cohort of patients, even if the data of the patients are distributed worldwide, and to launch very complicated classification, target, and biomarker discovery application on this cohort without moving data around. This intelligent cloud platform also capture and integrate knowledge coming from the scientific literature using its core NLP, natural language processing capabilities, and also integrate knowledge coming from public databases. And to date, we have knowledge coming from 120 databases and knowledge from the literature, and together with multi-omic data of patients' data, all this is integrated within our proprietary knowledge base that we call MemeSite. MemeSite is subsequently used through our patient GI2 algorithm, which is an algorithm that infer genetic interaction graph per patient. So this is an algorithm that uses multi-omic integration and systems biology, as well as supervised machine learning, to infer for each patient between 800,000 and 2 million genetic interactions, capturing the genetic relationship between genes in a given system. This could be the tumor, could be the immune system, if you're interested in autoimmune disease or if you're interested in oncology. So all this computing integration is totally automated, is integrated into our SaaS platform and fully accessible through a web interface by life scientists that do not need to know to code to use this system. So the graph of the patients of the selected cohort by the users are generated. And from this graph, because each graph is different, we're capturing patterns that allow the software to classify patients based on their graph. And to characterize, actually, the user can get extensive dashboards with characterization, molecular and clinical profile of each group of patients. Then we'll do also target discovery, indication prioritization. For clinical trial is actually a solution that enables actually to predict for each one of these graphs whether the patients will be a good responder to a drug, knowing only the name of the drug target. And the biomarker discovery will capture patterns with this graph to identify signatures that could discriminate good versus bad responders or patients with good versus bad prognostics, for example. So as I told you, we have a validated technology. We have shown that we have developed different algorithms using supervised machine learning that are highly performant to identify targets. So we have, for example, 5 times higher performance than classical approach to predict drug targets controlling metabolic reprogramming in triple-negative breast cancer. This is one use case that we got. We also shown 7 times higher performance than classical approach in predicting good responders to a new drug. And this is here shown in green in this graph in comparison to alternative approach looking at expression level of the target here for sorafenib in hepatocellular carcinoma. We've shown also more than 2 times higher performance of the signature identified using our biomarker discovery pipeline than other approach from similar profile that might use natural language processing or differential expression from different patients or cell lines. So in summary, with our solutions, you will have more than 2 times higher performance than competitive or solutions that are currently used in the industry. You will be able to do in 1 day what is usually done in 30 days by an internal bioinformatic or outsourcing service, and for 1/10 of the price. That allow you, if you have an internal bioinformatic team, to rationalize their time and to allow them to concentrate on strategic work, and also to empower 100% of your scientists that are usually mainly composed of biologists. This was a very, very brief introduction to our work. Please do not hesitate to contact me at sarah@memes.ai for any other information and to visit our website at memes.ai. Thanks a lot for that.
Hi, my name is Mike, and I am the founder of Neurotox AI. Our company was founded in 2020, and we are a team of professionals with extensive experience in regulatory toxicology and machine learning. With our diverse background, we were able to develop a technology that can accurately predict the toxicity of a chemical compound. In this presentation, I hope to give you a brief overview of how our platform can help your company to ensure drug product safety and reduce the likelihood of product recalls. The core focus of Neurotox is to leverage state-of-the-art machine learning methods to build explainable and accurate algorithms for toxicology evaluation. Computational methods have the advantage that they can process a large set of compounds at a time and return the results almost instantly. They also have the advantage of being able to make a prediction for unknown compounds as well, which significantly reduces the time and costs associated with toxicity testing. Neurotox offers two types of computational methods. The first is the rule-based model, which uses expert knowledge to identify substructures associated with a toxicity endpoint. The mechanism of action and mitigating factors provided by the model are all supported by the scientific literature and expert-derived knowledge. We also provide a statistical model, which uses machine learning method to predict the biological activity of a compound. When used together, these two models can complement each other and form robust and explainable toxicology predictions. Currently, one of the main use cases of our software is to identify and evaluate mutagenic impurities that arise from drug manufacturing processes. According to the ICHM-7 guidelines, drug companies are required by law to identify and categorize the mutagenic entities in their products to limit carcinogenic risk. By using the tools presented earlier, companies are able to comply with international regulations, ensure product safety, and reduce the likelihood of product recalls. We have several features that can greatly improve your regulatory workflows. First, our software is implemented as a web app, which can be accessed from anywhere and anytime. Second, as mentioned earlier, using both the statistical and rule-based model will allow your company to comply with ICHM-7 guidelines. The reports generated by our software can also be used as part of your regulatory submissions. Third, our algorithms have 100% chemical coverage, meaning that they're able to generate a valid prediction for any small molecule. Now I will hand it over to my colleague to go over a few case studies on how our software can be used for evaluating mutagenic impurities.
Hello, my name is Stéphane Gagné, and I am the president and CEO of Ovensa. Ovensa is a Canadian company working with the pharmaceutical and biotech clients to increase the efficacy and reduce the toxicity of their drugs using our patented drug delivery platforms. We have deep expertise in making nanoparticles using our biopolymer that provides a complementary approach to lipid nanoparticles. We specialize in the formulation of siRNA, mRNA, and other RNA therapeutics to bring them across the mucosa or the blood-brain barrier. We have also developed formulations to avoid or minimize delivery to the liver. For those who are familiar with chitosan, our trimethyl chitosan, that we call also triozan, provides many benefits such as solubility in water and at a wide range of pH. We have successfully formulated many drugs that were insoluble in water. Our trimethyl chitosan is patented. It is biodegradable and highly stable, even beyond 100 degrees Celsius, which opens many possibilities for its use and for manufacturing. The main characteristics for triozan is that its cationic charges allow for opening the tight junctions and cross the barriers. So this is why we use it to make nanoparticles in order to protect the drug and deliver it more specifically to certain tissues or cell types using ligands that can be conjugated to the triosan for targeted drug delivery. Another advantage is that it can be used for various routes of administration, such as intravenous, intra-arterial, intranasal, subcutaneously, or for oral delivery for adherence to the wall of the small intestine. Those are examples of formulations that we have done using the triosan nanoparticles without any conjugated ligands. We have formulated with siRNA, mRNA, antibodies, large peptides of up to 400 kilodalton, and also small molecules. Those were made for different routes of administration and for various types of diseases, most of them for brain delivery. We have also prepared nanoparticles with conjugated materials. In addition to the triosan biopolymer to make nanoparticles, we have also a bradykinin 1 and bradykinin 2 receptor agonist that we use for further increase the delivery of drugs across the blood-brain barrier. This is a good example of such potential of the B1R and B2R agonist that we have as tools to further increase the power of our triosan nanoparticles to deliver cisplatin to the brain and the surrounding tissues using a brain cancer model. In this example, we show that triosan alone, without agonist, is able to significantly increase the delivery to the tumor without affecting much the delivery and clearance by the liver or the kidneys. Considering the interest for the delivery and increasing the stability of mRNA COVID vaccines, as well as siRNA drug candidates, we've prepared mRNA and siRNA formulations using our triosan platform and shown that we greatly improve the stability of the siRNA once stored at -20 degrees Celsius following flash freezing with liquid nitrogen, compared with the siRNA alone that are not suitable if they are not protected by the triosan nanoparticle. We then wanted to compare such stability during storage at -20 degrees Celsius with the free siRNA and free mRNA for a shorter period of time to see how soon the non-protected RNA degrades. And you can see that both RNAs are quite stable if they are in our nanoparticles, but not as much if they are formulated or alone without our protection, if they are without the nanoparticles. So really in this case, the triosan nanoparticles greatly increase the stability of the mRNA payload. We finally have looked at the stability of our siRNA nanoparticles, not only at -20 degrees Celsius, following a flash frozen procedure, but also at 4 degrees and 37 degrees Celsius. And we have noticed that we can also protect it at at various temperatures, although the stability obviously starts to diminish at 8 hours. In terms of safety and biodistribution, we have demonstrated the safety of our triosan nanoparticles for the delivery of our siRNA to the tumor in a head and neck cancer model. We also see the delivery to the kidneys and lungs, but not in the liver or spleen, which suggests a longer circulation time and reduced mononuclear phagocytic system effect. This slide shows at the bottom left that we can bring a siRNA drug candidate to the brain with good accumulation in the tumor. On the upper right side of the slide, right here, you can see the benefits of the triosan delivery system with a good accumulation of the siRNA in the olfactory bulbus and excellent retention over time, which is very important for allowing the passage to the brain. And at the bottom right here, we have similar results with an antibody that on the left is cleared quite rapidly by the mucus if not protected by the triazan. But on the right, the trimethylchitosan or triazan and its positive charges have a strong adherence to the mucosa and allow for delivery to the brain, whereas the antibody alone cannot make it to the brain. So this is with the triazan, and this is without the triazan here. So in summary, our triazan platform can be used for formulation purposes to protect your drug payload using a non-lipidic approach. And we can also use ligands for more specific delivery to certain cell lineages or tissues. And we have also tested additional properties of triazan for fighting antibiotic resistance or to dissolve the aggregation of the beta amyloid or tau protein aggregation that are known contributors in Alzheimer's disease. We can count on an international scientific advisory board with 3 oncologists and experts in RNA therapeutics, formulation for brain delivery, and targeted drug delivery. Ovensa is currently very active to identify potential acquirers for M&A or for strategic alliances that will allow us to serve more pharmaceutical and biotech companies. So if you are interested in exploring this platform for your drug candidates, for a potential licensing agreement or M&A transaction, please contact me so that we can explore how we can work together. Thank you.
Thank you for your interest in learning about UHN. Headquartered in Toronto, Canada, one of the most multicultural and cosmopolitan cities in the world, UHN is Canada's leading academic, teaching, and healthcare research network that serves over 1.2 million patients per year through its vast network of clinical programs. With a rich history of world-class discovery and innovation, we are inviting you to gain access to UHN's enterprise to access knowledge, leading-edge technologies, and expertise to accelerate the achievement of your research goals. On to the next slide. UHN's research discovery journey started in 1922, when our investigators led the world's first clinical use of insulin. From there, research innovation has thrived over the years, including the discovery of blood-forming stem cells in 1961, the clinical world first single and double lung transplants in 1983, and the single liver cell map for regenerative treatment in 2018, to name a few. UHN is home to Toronto General Hospital, Toronto Western Hospital, Toronto Rehabilitation Institute, the Princess Margaret Cancer Centre, and the Michener Institute of Education, Canada's only academic institution devoted exclusively to applied health sciences, education, and the Institute for Education Research. UHN research institutes and hospitals work together to advance science. This has positioned UHN as Canada's premier academic medical center with major programs in cardiology, oncology, transplantation, and many other disciplines. On to the next slide. By aligning resources across hospitals and research institutes, UHN has enabled clients to access an extensive range of research services. As such, UHN provides state-of-the-art equipment along with techniques, training, and management of research projects that meet the demands of any academic or industry team. On to the next slide. UHN's multidisciplinary team carries out a wide variety of services. UHN's unparalleled expertise in clinical research represents a pillar to offer unmatched services in a wide array of scientific fields. Today, we are offering a gateway to access services to support rehabilitation studies by accessing UHN's simulation laboratories, services to manage multi-site clinical trials, biospecimen services, access to UHN's multi-center imaging infrastructure and technologies, imaging services to support your clinical trials, and state-of-the-art simulation labs for testing medical service devices. On to the next slide. UHN rehabilitation simulation labs enable the study of human abilities and performances necessary to validate new products and technologies focused on improving the well-being of people. The state-of-the-art simulators enable researchers to use hydraulic-powered platforms and virtual reality technology to measure day-to-day hazards in a safe setting. The STAIR Lab assists research teams to determine ways to improve mobility independence and prevent injury through GATES. This success has led to the adoption of informed clinical recommendations, the adoption of building codes, and safety standards. The Driver Lab is the most technologically advanced simulator in Canada and is among the most sophisticated driving simulators in the world. The lab explores the impacts of health and medications on driving performance as a means by which to increase driver safety in healthy older adults and those living with injury or illness. The Street Lab produces a 240-degree projection of a busy street through the eyes of a pedestrian. Persons with hearing loss are 3 times more likely to experience a fall. Within controlled and repeatable virtual reality environments such as this, research teams are able to precisely analyze when and how daily tasks such as walking can trigger a dangerous fall. The Winter Lab operates as an environment within which researchers may develop and test new winter gear and products, such as footwear or clothing, to define mechanisms to enhance performance on icy and snow-covered surfaces. On to the next slide. Focusing on your needs, UHN provides comprehensive clinical research services to support your preclinical and clinical studies. Providing a full range of support services for a wide variety of study types, we have become a highly sought-after partner for both large and small corporations and academic institutions. We provide quality services to meet the demands of today's complex drug development process. From the initial study design to the final regulatory submission, UHN will ensure you reach your development milestones in the most efficient way possible. On to the next slide. With over 20 years of clinical research experience and dedication to the highest standards of quality, UHN is a reliable partner for academia, diagnostics, and biopharmaceutical companies. From small and simple projects to complex prospective studies, UHN has the expertise, inventory, and robust clinical networks to meet your biospecimen needs. Besides offering biospecimen storage and curation services, some of our services include high quality, well-annotated solid tissue and biofluids, tissue microarray construction, among others. On to the next slide. Our multi-center imaging laboratories provide end-to-end de-identification, data transfer, testing, and analysis support for multi-center clinical trials. Our clients benefit from improved consistency and reliability in clinical trials imaging data. Among the services offered is the utilization of a safe testing environment that includes data storage and computer power to validate imaging AI solutions. Among other services listed on this slide, our laboratory also supports theranostics and molecular imaging studies. On to the next slide. As the largest medical imaging department in Canada, our multi-center imaging laboratory is anchored in the expertise of a multidisciplinary team of innovative technologists, nurses, and radiologists. UHN offers to you our expertise in AI technologies and translational research to support studies with exceptional image quality and analysis. Our services include tissue biopsies for clinical trials, tumor measurements, medical image anonymization, and high-fidelity data transfers. On to the next slide. As a world-renowned human factors center, UHN supports pharmaceutical, medical technology, health IT, and healthcare organizations, as well as government agencies in the design and testing of health products and services, addressing the complexity of human behaviors. Our simulation labs enable clients to conduct testing on medical devices, digital therapeutics, healthcare environments, and innovative service delivery models. Some of the services offered by our human health factor laboratories include the conduct of observational studies, contextual interviews, and the conduct of environmental scans, and many others. On to the next slide. In closing, we hope you enjoyed our presentation and invite you to visit our website at researchinnovationcores.uhnresearch.ca to discover the research solution that meets your demands. It will be a pleasure to work with you and help accelerate the achievement of outcomes of your research studies. Thank you.
Hello, my name is Handel Kim, and I'm the CEO of Variational AI, and it's my pleasure to introduce our company to you today. At Variational AI, we're building a discovery engine that will rapidly generate novel and optimized small molecules for unmet medical needs across disease areas to redefine the economics of drug development. We call our discovery engine ENG, and we're starting with kinases. We've chosen kinases as the initial target class for the reason that there is relatively de-risked biology. We are going after validated targets, and there are 71 FDA-approved inhibitors across various kinases. As we are an AI for drug discovery company, we need data. And there exists a massive amount of data, thousands of kinase crystal structures in PDB, thousands of unique ligands and trials with kinase inhibitors in the public domain, as well as this significantly large set of data available commercially and much more that can be generated. Selectivity amongst kinase receptors is also a major concern. Some drugs fail for off-target effects, while others actually benefit from hitting multiple synergistic targets. Our ability to control selectivity in the generation of novel molecules is a key differentiator of variational AI. When we look at the landscape of approved kinase inhibitors, and we look at the 110 novel kinase targets that are under clinical development, we notice that there is a lack of diverse scaffolds. They seem to be clustered amongst these classes of scaffolds. But of course, there is significant structural similarity across these kinases, which may explain the reason why there's clustering. And in fact, the ATP binding site is conserved across the entire class of kinases, and which leads us to go after orthosteric kinase targets. Training ENG on a panel of many such kinase targets increases the amount of already substantial training and also allows us to leverage transfer learning. What we are building is a unified kinase model that leverages transfer learning. We train on, as we mentioned, public and commercial data sets, as well as data generated in silico. We select a number of kinases to train, and we put that through our algorithm ENG. And what we do is we generate very rapidly with a unified model in a matter of weeks, potentially per target, novel and optimized leads for a number of kinases. And of course, we are going after oncology and specifically focusing on on solid tumor. At the heart of our differentiation is generative AI. The difference between generative AI and the status quo is when we look at the status quo and we look at chemical space is in a massive and chaotic search space with some estimates of 10 to the 60 molecules that exist in chemical space, which is an absolutely terrifying number, almost equivalent to the number of atoms in the visible universe. More importantly, it's very chaotic. Navigating by structure across chemical space is a not fruitful approach in the sense that a small change in structure of a molecule can induce a very large change in property. It's impossible to effectively navigate the full space using brute force methods. By contrast, what we do is we project structures into an ordered space known as a latent space, where we reduce dimensionality, and then we do a simple gradient optimization within the latent space, and then map back out into structures. What we do is we use a continuous representation of an otherwise discrete landscape of molecules. The optimization, therefore, is efficient and data-driven. We started the company in 2019 with state-of-the-art machine learning, where we outperformed benchmarks in the machine learning community from leading labs at MIT, Stanford, and entities such as Google and Benevolent AI, as well as Genesis Therapeutics. And we started from this and have built and changed and completely and qualitatively improved the predictive power of our generative AI model and are taking it out into active development. We have collaborated with a number of entities across disease areas where we've proven the multi-parameter optimization, where we've been able to generate novel chemotypes for very low data targets, and to leverage transfer learning to accelerate the development of therapeutics. We are based in Vancouver, Canada, founded in 2019, with a leadership team composed of top machine learning expertise, as well as extensive drug discovery success. Our machine learning scientists are truly scientists from leading institutes such as MIT and Caltech, and from companies such as Microsoft, Google, and D-Wave. On the drug discovery side, our leadership team has over 124 patents, has taken 11 drugs into late-stage clinical trials, and multiple drugs licensed to pharma. We have raised seed funding from leading investors in the United States and Canada, and also have attracted non-dilutive funding from government funding agencies. We are focusing our unified kinase model on solid tumor indications. ENG is able to generate novel leads optimized for multiple properties in weeks to 2 months. We're very interested in working with companies seeking novel kinase inhibitors for partnered programs or licensing. If you have any questions, please feel free to contact me. Thank you very much for your time.
Hi, my name is Mike, and I am the founder of Neurotox AI. Our company was founded in 2020, and we are a team of professionals with extensive experience in regulatory toxicology and machine learning. With our diverse background, we were able to develop a technology that can accurately predict the toxicity of a chemical compound. In this presentation, I hope to give you a brief overview of how our platform can help your company to ensure drug product safety and reduce the likelihood of product recalls. The core focus of Neurotox is to leverage state-of-the-art machine learning methods to build explainable and accurate algorithms for toxicology evaluation. Computational methods have the advantage that they can process a large set of compounds at a time and return the results almost instantly. They also have the advantage of being able to make a prediction for unknown compounds as well, which significantly reduces the time and costs associated with toxicity testing. Neurotox offers two types of computational methods. The first is the rule-based model, which uses expert knowledge to identify substructures associated with a toxicity endpoint. The mechanism of action and mitigating factors provided by the model are all supported by the scientific literature and expert-derived knowledge. We also provide a statistical model, which uses machine learning method to predict the biological activity of a compound. When used together, these two models can complement each other and form robust and explainable toxicology predictions. Currently, one of the main use cases of our software is to identify and evaluate mutagenic impurities that arise from drug manufacturing processes. According to the ICHM-7 guidelines, drug companies are required by law to identify and categorize the mutagenic entities in their products to limit carcinogenic risk. By using the tools presented earlier, companies are able to comply with international regulations, ensure product safety, and reduce the likelihood of product recalls. We have several features that can greatly improve your regulatory workflows. First, our software is implemented as a web app, which can be accessed from anywhere and anytime. Second, as mentioned earlier, using both the statistical and rule-based model will allow your company to comply with ICHM-7 guidelines. The reports generated by our software can also be used as part of your regulatory submissions. Third, our algorithms have 100% chemical coverage, meaning that they're able to generate a valid prediction for any small molecule. Now I will hand it over to my colleague to go over a few case studies on how our software can be used for evaluating mutagenic impurities.
We will briefly demonstrate case study examples of when a company submits a compound using our software and how to interpret these results to come to various conclusions. Our first example is 2,3-methylphenoxymethyloxyrane. This compound was predicted to be positive by statistical and rule-based model. The alert flagged by a rule-based model was glycetyl ether, highlighted in red. Glycetyl ethers are strong electrophiles that can react with nucleophiles, such as DNA. If we further analyze the data, our training set examples show similar analogs from our statistical model. Through the implementation of a read-across approach, these similar analogs were known positives in the Ames test, and thus the query structure was concluded to be positive. Our second example is an oxa-aza-tricyclo-no-name compound. This compound was predicted to be negative by the statistical model and positive in the rule-based model. The alert flagged by a rule-based model was an epoxide highlighted in red. One of the mitigating factors of epoxides is that they are susceptible to steric influences. If we further analyze our training set examples that show similar analogs, these similar analogs are no negatives in the Ames test, and using a read-across approach, the query structure was concluded to be negative. In addition, our rule-based model has in-depth scientific explanations of the alert structure, sourced through literature, which facilitates easier expert reviews for final decisions. To conclude, our software can quickly and effectively facilitate the assessment of mutagenicity, a method recognized and accepted by regulatory agencies under ICH M7.
Thank you for listening, and I hope you found this session useful. If you have any questions, please visit our website or contact me via email. Have a great day. Hi, my name is Mike, and
Thank you very much. I hope you had very good information. That was Canadian companies. Thank you very much for watching. So, ladies and gentlemen, if you have any question, please send a question to the presenter online. You can scan the QR code or access via live on the homepage, and the presenter will choose the questions and answer to you. Once again, thank you very much for your attention. And this concludes the BioKorea 2022 morning presentation session. And the 5 companies will be presenting starting 1 p.m. So I hope to see you again at 1 p.m. Korean Standard Time. Thank you very much.
Better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got it.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got it.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got it.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got it.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got it.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got it.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests,
Don't worry, you got this.
more effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day. For the good of millions all over the world. We rise and get to work. So we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines. And life-changing breakthroughs. And we never stop in pursuit of answers. Every single day. For the good of millions all over the world. We rise and get to work. So we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines. And life-changing breakthroughs. And we never stop in pursuit of answers.
네, 안내 말씀을 드리겠습니다. 바이오 코리아 2022 바이오 라이브 이제 오후 세션을 곧 시작하도록 하겠습니다. 부산에 계신 여러분들께서는 이제 라이브 존 안으로 들어오셔서 착석을 해주시고 함께 해주시면 대단히 감사하겠습니다. Ladies and gentlemen, may I have your attention, please? Bio Live will be starting momentarily, so please enter the live zone and be seated. Thank you. Welcome back from your break, ladies and gentlemen. I hope you enjoyed the lunch break. In BioLive 2022, we provide you insights on the current status and prospects of the global biohealth sector. In day 3, we also have very prominent companies which introduce their technology and product. You can definitely look forward to it. So in the afternoon session, first, Mr. Cresswell, the Vice President of GoCuba Technology, will present first. So without further ado, let's welcome him onto the stage.
Thank you, everybody. Thank you. Yeah, so I guess I can start right away, right? Yeah. So yeah, hope everybody had a good break. I had some tasty food downstairs in the mall. Some really great food here. So yeah, my name's Leland Cresswell. I'm the VP at GoCuba Technology, and it's very nice to have your time today to listen to my presentation. I'm sure you've probably listened to a lot of presentations here, so I try not to waste your time too much, okay? So I guess just to start off, I'll speak a little bit simply about what we're doing. We're trying to do digital therapeutics, which mean using digital devices and digital technologies to try to help people recover from sickness. And specifically for us, we look at vertigo. So vertigo can come from many different causes, but it basically means that you have a problem with dizziness and the inability to maintain your balance. So I think that most of the people here are pretty young, so you haven't, I guess, encountered yourself probably vertigo in the extreme, maybe sometimes feeling a little bit dizzy. But when we're older, the prevalence of vertigo as an actual disease and a chronic illness is actually quite high. I think you might be surprised. And imagine in the day, if you have vertigo 2 times or 3 times for 10 minutes or 15 minutes each, it can be very difficult to do work, it can be very difficult to focus on anything, very unpleasant. And this is a chronic illness that happens over and over again. And it's also difficult to treat. So what we're trying to do is we're trying to use new digital technology and mixed reality to help people with this illness. So the current issue in this market and around vertigo is that it's quite expensive and very time intensive to help correct problems with vertigo. So if you see here, I guess I have to speak here. I'd like to walk around, but can I grab this? Is it okay? Yeah? I won't try. But you have to usually be beside the doctor, beside the specialist, working together with them on either a daily routine or in some cases just a weekly routine. So that takes a lot of time from a specialist doctor. So you can't just have any general doctor doing this. It requires a specialist doctor. And so these individuals, of course, they have special training, and it's also not cheap to have them work with the patients, but they require it. So there's a big cost problem here in this market. And so that's one of the things that we are trying to solve with our solution and with digital technologies. And then the other problem is that there are many therapies, widespread therapies, especially in developing countries, where the therapies are not very effective. So here in Korea, we have a very top-tier healthcare system, I think. So the therapies used in Korean hospitals are very effective. But if we go outside of Korea, we find many countries and many areas where the therapies are not very effective. And in addition, there's also many countries and countrywide healthcare systems that don't use digital technologies. It means that the data and the proof of if someone is getting better, how they're doing, this proof is very difficult to grab because it's all in paper and you have to actually go out and try to find it if you want to know how the person is doing or if you want to do a research study. Many countries outside of Korea are not using, again, digital systems for tracking this data. It's another big problem. So, to speak about what we're specifically doing, so mixed reality, we focus on the software side of the solution. So the hardware side for mixed reality, I think in this exposition, there's I haven't seen any companies actually using mixed reality. There might be one or two if you walk around and look. But mixed reality is still a very, very new market in terms of what consumers can use. VR, virtual reality, is, I think, growing very quickly, and it's a little bit more mature. I think because there's more applications to general consumers in terms of games specifically. Mixed reality is much more useful in, I think, business and healthcare. So it's a little bit slower to grow, and it's also, the technology is not as easy as VR, because in mixed reality, we have to have the physical reality mixed together with the virtual, which is actually quite difficult, especially in terms of the hardware, but also in terms of the software. So here you see a Microsoft HoloLens 2 device. So this is the second generation device, but it's still early days. So this is one of the main devices that we use, and we put our software on top of. And specifically what we do is we get data from how the person is moving their eyes and how the person is moving their head and body. All of this data is being tracked by the HoloLens 2 device, as well as the other mixed reality devices we use. Because mixed reality, you have to know how the person is moving for many different applications. So what it means is these devices that we are running on top of also happen to be much, much cheaper than specific hospital-grade devices that 10 years or 20 years ago were being used for the same thing. So 10 years ago, if you need to do eye tracking or body and head movement tracking at a hospital, you're looking at hardware that's costing $30,000, $40,000, $50,000 or more. So again, the problem of cost is something that now we're able to solve with this technology. And so specifically our software, we are able to recreate many of the physical exercises that the doctor does with the patient. We recreate that in a mixed reality environment. And the big benefit of this is twofold. First benefit is the doctor doesn't have to be standing beside the patient all the time. And the doctor doesn't have to be guiding the patient all the time. So what this does is, previously, without this technology, a doctor would spend 1 hour together with the patient, very, very focused on the patient, because he also has to make sure that the patient is moving correctly. Because in terms of vertigo therapy, the movement of the patient and the way that they're doing the exercise is very important. I'm sure many of you have gone to a physical fitness center, a gym, maybe for building muscle or cardiovascular, right? It's very important to do those exercises correctly. Otherwise, you can hurt yourself, right? It's very similar in terms of vertigo therapy, but it's even more sensitive. Okay? So instead of having the doctor beside the patient 100% of the time, doctor only needs to be beside the patient maybe 20% of the time or 30% of the time. The remainder of the time, they're using our device, which is tracking their body movement very, very specifically and making sure that they are doing the exercises correctly. So the end benefit for the clinics and the hospitals and the doctors and the senior rehabilitation centers that we work with is about a 60 to 70% reduction in the cost of labor, the cost of specialist time that is achieved with our software. Okay? So this is an overview of our business. As I said, we have exercises, different routines that the patient would go through. We collect data. The data is used to make sure they're doing it correctly, and the data is also used later on by the doctor to track the progress. Again, tracking the progress of the person is very important. Because this device can be used remotely, we can also have the patient at home doing 30 minutes every day very, very specific exercise routine, very specific therapy routine, and all of that data about how their body is moving is being processed by our AI and our machine learning technology, and we're giving that straight to the doctor so that they know specifically what the progress is of the person. So in the past, it's not possible to say to a patient, go home for a month, do these exercises and come back, because the doctor would have no idea if the person is doing those exercises correctly. He wouldn't have any data. He wouldn't be beside them to guide them through it. Totally impossible. But with our device, remotely, this sort of thing is now possible. I'm sure that maybe some of you have also kind of seen this effect in terms of the COVID issue here in Korea and also globally. Because of that, we see that remote technologies are actually very valuable, especially someone who is quite sick with COVID. You don't want them to come to the hospital immediately. Ideally, maybe you can do some initial screening while they're at home, if it's possible for them to stay at home. So I think that through COVID, we've seen that remote technologies can actually be quite beneficial. Because I know that in Korea specifically, before COVID, remote doctor care in many cases was illegal and not well accepted because of, I guess, a fear of misdiagnosis and some other reasons. But now, and I hope through our solution as well, we can show people that we can be helping people in a remote way that's also effective. So here is just a quick video of a very simple exercise, just a walking exercise. And the person walks down the mixed reality street. We're getting information how their body is moving. And then they walk back. So even just a simple exercise like this needs to have information about the patient. Are they walking straight? Is their body falling to the left? Is their body falling to the right? These are the reasons why a specialist needs to be there previously. But now, while they're doing this, we're getting the data and we're processing it for them. So a little bit about our specific company. So we're a startup, and we're working out of Seoul Biohub. And this device, this software that we're building, will be classified as what's called a Class 2 digital therapeutics device. That's a Class 2 in the FDA and also KFDA. So, of course, we want to be a part of the actual healthcare system as well, which means we require a regulatory approval, FDA, KFDA. And as part of that, our company has been preparing itself through a few different ISO certifications that are required for this. In terms of the sort of partnerships we have and who we work with, so Cha Byung-won is a Cha Bundang Byung-won, is a very important partner with us. We work with them on our technology. They're helping us to understand how exactly we need to process the data. And then in terms of our beta partnership and getting our technology out to people, we work with Seongnam City. So Seongnam City gives us support, and we are trying to bring our technology to the seniors there and people who have issue with vertigo. So as I said, there are virtual reality companies that do this sort of thing. But the big difference between mixed reality and virtual reality, especially for older folks, is that virtual reality can be very difficult for them to use because it puts them into a completely different environment that's completely blocked off from outside. However, with mixed reality, we can still have people nearby, maybe helping them, and they feel much more comfortable because they're in the real environment, right? And so we're a startup, right? So for us, it's very important to build something that we can commercialize and that we can actually sell and go to market. If if no one wants to use our device, then even if our algorithm is very accurate, and even if we have a good effect at helping rehabilitation, nobody will use our device, right? So with mixed reality, we believe that we can build something that, especially seniors, people who are 60 plus usually, who are actually having problems with vertigo, that they would actually want to use our device at the senior centers, at the clinic, and to enjoy it and to not say to the doctor, oh, I don't want it, I don't want it, I hate it, it makes me dizzy. We have to build something that we can sell, and mixed reality is the best way to do that. And as I said, there's not a lot of companies who are using mixed reality right now. It's a new market, and so we're trying to get ahead and be a first mover here. The market is growing very quickly as well. I'm sure many of you know, of course, here in Korea, the number of seniors is growing very quickly. But digital therapeutics as well is, especially, again, due to COVID, is growing very, very quickly. So, this is, we believe it's a good place to be and a good place to make impact on people's lives and on improving rehabilitation and choices for patients with vertigo. So our company, I guess just to briefly conclude, again, we're a startup company. We're doing a clinical trial this year. So we are looking for partners who might be interested in commercializing our software in Korea, but also in other markets, maybe next year. And then we're also looking for investment into our company, hopefully later this year or early next year. So this is kind of just our roadmap. Again, next year we're hoping to do a pilot beta program and to commercialize our software and our technology. And then hopefully go abroad as well, which is not so easy as a Korean company, but hopefully get traction outside of Korea as well. So thank you for listening to my speech. I hope that today I brought some information about our company that might be useful to some of you. I hope I didn't waste too many of your times. If you have any questions, I think we have a brief Q&A time right now, so please feel free.
Yes, thank you very much for that excellent presentation. As you mentioned, that we have about 10 minutes for Q&A, so just feel free to raise your hand and give us a question. Please, we'll get your microphone there.
Hello. Thank you for the presentation. I have a quick question. So I am a user of VR, so I understand that these headsets can get pretty heavy. And
oh, yeah.
Yeah. And as you said, like since your target is the senior
older
population,
folks, yeah.
I'm wondering if you found any difficulties of the weight affecting their balance and vertigo as well.
Yeah, no, that's a really great question. We were actually talking about that yesterday, just me and some of the team. So there's two major devices that we're working with right now. Again, like our value is around the software. So as the mixed reality market grows, we have more options. The one I showed you, the HoloLens, is comfortable enough for a short 30-minute or so exercise. So if we need high accuracy and a short exercise, like at a clinic maybe, or at a senior center, then the HoloLens device is really great. But if it's something that we want them to use it for an hour, or we want them to use it like daily, then there's another device called the ENG device. I don't have any pictures in the slide deck, but I guess you can come over to our booth over at J6, and we have two of the devices there available to show you. But they're almost like sunglasses. So I can't remember the exact weight, but they're much, much, much lighter than a VR headset would be. They basically look like sunglasses, but they also have the critical cameras and the small bit of processing that's required to overlay the images. So those are actually quite comfortable. But yeah, it's a huge problem. That's a huge problem. We've seen many other technologies, especially here in Korea, that have in the past tried to do something with vertigo and dementia as well, in terms of VR. But the issue is after the clinical trial or even during the clinical trial, no one wants to use the device, especially the seniors, right? They don't want to use it. Maybe the content is kind of boring, or in many cases it's just very heavy and disorienting. Yeah, so for us as a company that needs to try to build something we can sell, that's really important. Thanks for the question though. That's, yeah.
Oh, thank you for the answers.
Thank you very much. Any other questions from the floor? If there are no more questions here, then thank you very much, Vice President, for that presentation.
Well, thank you very much, everybody.
Thank you. Or you can send a question to the presenter through online. So you can visit our official homepage and leave your question. Or you can scan the QR code. You can visit there. Thank you very much. So moving on to the next company, I'm so excited to introduce the next company called Protina. And I would like to invite the founder and CEO of Protina, Mr. Tae Young-yoon, onto the stage. Please welcome him.
Hello, ladies and gentlemen. I'd like to send my sincere thanks for introducing me kindly, and also my thanks go to organizers for giving me this opportunity. So Protina is actually provide the diagnostic device for molecular materials. COVID-19 made a lot of changes in our lives. So we have the various molecule-based diagnosis experiencing COVID-19. So we have a simple molecular diagnostic devices, but when we see oncologists, and there are quite the complex data and information, and based on the analysis of those informations, we can provide target treatments. So my company is targeting this the molecular diagnostic market. So this is the prediction that the molecular diagnostics market will increase at the 10% every year. But experiencing COVID-19, we are expecting much faster growth as the biosensor and decisions and the lawsuits and those the molecular diagnostic the companies are providing the solutions. So the Protina is a small company compared to those the big movers and shakers. So what can be offered by my company? So actually we have a differentiating factor, which is PPI, the protein and protein interactions. The inside our body, the proteins and play very important role. But proteins have the interactions that they actually have a kind of the clusters in order to have the actions inside our body. So this is the membranes and eggs, and the signals can be delivered in this way. So you can see all these small arrows, and those are based on the protein-protein interactions, which is a TTI. TTI is crucial inside our body in order to deliver the messages and signals. So in the past, the drug development was made in order to block this PPI. But in the future, the study will be focused on the PPI inhibitors and also the PPI promoters. So Penkelexter also used this the PPI, the inhibitor. PPI inhibitor need the molecular diagnostics. In order to do that, it is important to figure out what is the amount of that protein. But the NGS technology is available that was developed about 20 years ago in order to assess the amount of the DNA. But when it comes to the amount of PPI, we do not have available technologies. So what I want to say is, when I see universities and research labs and the doctors, collect about 10 or 15 PPI. The Westblon technology is used in order to analyze the PPI, but it took quite a considerable time. It was difficult to apply in the clinical trial because it took considerable time. But all the data can be integrated on one single platform based on the technologies developed by Protina. That is the way to go. So let me explain more about PPI, how the clinical trials can use the protein-protein integrations and how the measurement can be done in accurate manner. So blood can be used and other tissues can be used in order to check a PPI. So PPI complex can be extracted without damaging that, and that will be placed on the substrate. The antigens will be placed, the substrate, and extracted materials will be injected here. So the target proteins will be analyzed in this way. But there are more than 20,000 proteins. So in order to reduce the level of noise, and we only focus on the targeted the proteins. So we successfully attached these materials to target protein, so we could increase the accuracies while reducing the level of noise. This is kind of the PKI complex analysis technologies and also target proteins, and can be analyzed based on the PPI the connections. In order to analyze the amount of protein which are not connected to the materials, we could have the additional analysis. So antibodies and all these drug materials should be packed and should be matched together. So this can be the image of the combinations and of these integrations. In the past, in the clinical trials, it was difficult to have exact result and the data about the PPI. And this is actual images. This like a star on the dark sky at night. So this is to show all the detections of all the PPI. So there are a few important factors to increase the accuracy. First, it is important to have the PPI complex from tissues and blood. But as I mentioned, this is the technologies to collect the PPI in intact way without damaging these proteins. When we see stars, and if we go to the areas with the many light, then it is difficult to see the stars on the night sky. So same thing goes here. So if there is a high level of noise from other kind of protein, it is difficult to have the targeted the performances. So we select a few targeted protein, the targeted PPI, so those will be targeted in exclusive way. So when we see the conventional technologies, so it is difficult to see all the holistic views. So, well, that looks like kind of a Milky Way. And let's say the star, there were about 100 stars, but when the number of stars go up to 150 and 200, it will be much difficult to select and find the targeted the stars. So we have the additional technologies in order to have a better target. So single molecule co-IP platforms and single molecule countings and superior surface quality are three differentiating factors. So in order to find out the targeted protein in a more accurate way. So there are a few chambers, so proteins can be induced to this chamber, so produced and then processed chips will be inserted in this chamber without the extensive trainings and analysis can be done. This the device actually get the focus on the targeted protein. By analyzing this protein, the PPI result can be produced in a fast and accurate way. So we successfully established the platforms to analyze this. So regarding these technologies, we are global top 5 company. Regarding the solid tumors and oncologies, and we have a great advancement indeed. So we have our IP regarding these technologies. So NK, the BCL2, the analysis was done. So we developed the platform for this. And after having agreements with big pharmas, the last year we conducted additional studies. So regarding the blood cancers and the specific PPI, the compound is reserved. That was the finding we had last year. So now this drug will go into phase 1 and phase 2A, the clinical trials. The big pharmas actually create the concerns about the applications, and the drug can be applied to patient, but what can be the dose? So those escalations and those the translation is very important. So FDA had the Optimus the analysis requirements. So the farmers had the dose analysis in order to receive the approvals, but there are a few the A cases, the severe the AE cases, and also the farmers actually had the concurrent the drug applications, but that had another side effects and adverse effect. That's why FDA is requesting please provide a minimum level of dose that can be applied to patient. That's why we had an additional study. So by using this platform, and we can figure out the minimum the dose levels. So farmers actually send the samples, so we can analyze that one and offers the analysis data, which shows the optimum and the minimum level of dose that can be applied to patient without a DAE. Regardless the locations, whether it is analyzed in Seattle and Seoul and in other cities, the analysis can be done. And in order to have the clear pictures, it is important to combine all this data. And we want to have an international standard in order to analyze data. Then we can better prove the meaningful the result of this analysis. When the optimum level of dose is used without big and severe adverse effect, and that this drug can be well studied into the later and advanced at the stage of clinical trials like the phase 3. So this data is crucial. It is important to develop the AC. But currently, the pharmaceutical companies have great challenges now. Sometimes the drug, the study drug have the efficacy, but sometimes in order to have a high level of efficacy, this study drug should be used together with the chemotherapy. But the case by case, the result is different, the response is different, and the behavior of this study drug inside the human body is different. That's why all these different PPI complexes should be studied in very detailed way. So profiling should be done for the PPI. So the pharmaceutical companies have all the data. So some the subject have the efficacies based on the certain amount of dose, and certain the subject have the efficacy when the concurrent and the medication is used. All this data is secured from the clinical trials. So currently we plan to have the joint study in order to further study these results. So we are going to have the further study for the head and neck cancers and breast cancers, and there are many different kind of the oncologies, and so we are going to expand our indications. So, in summary, when the pharmaceutical companies are developing new drugs from the end to end, we can provide the necessary support. So, NGS, the based analysis is done by many companies, and the amount of protein is analysis by many companies. But well, there are not many companies that can analyze the PPI. So in that sense, the Proteina is one of the companies that can offer such as the specialties. So PPI can be used. Analysis of a PPI in the range of 50 and 100 can be done. Based on that, pharmaceutical companies can have a better understanding about the characteristics of the protein. So in the clinical trials, the dose level of a study drug can be the study. So study can be designed in optimum way, and also predictable biomarker can be developed further in the future. Then, as a DCDS, we can generate the continuous and the profits based on this. In the Anyang City Knowledge Center, and we are building the facilities. So the equipment and facilities will be established there. And based on the collaboration with big farmers, the technology has been matured. So at the end of this year, we are going to have a sales office in the United States. We are going to develop the more collaboration cases. So this is all about my presentations. So I talk about protein interactions, PPI. Based on that, drug development programs can be supported. Thank you for listening.
Thank you very much for your presentation. Since we have a full of audience here, so if you have any questions, please raise your hand. Then we'll get you a microphone. And for our online viewers, also you can send a question to the presenter online. You can scan the QR code or access via live on the homepage. So we look forward to your participation. So any other question from the floor? Okay, then this is the end of Proteina. Thank you very much. Thank you. That was full of useful information. Okay, thank you very much for joining us. And next presentation will be starting at 2 p.m. in Korean Standard Time. Thank you very much. I'll see you soon. Thank you.
Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day. For the good of millions all over the world. We rise and get to work. So we can help bring you better tests.
Don't worry, you got it.
More effective treatments and vaccines. And life-changing breakthroughs. And we never stop in pursuit of answers. Every single day. For the good of millions all over the world. We rise and get to work. So we can help bring you better tests.
Don't worry, you got it.
More effective treatments and vaccines. And life-changing breakthroughs. And we never stop in pursuit of answers. Every single day. For the good of millions all over the world. We rise and get to work. So we can help bring you better tests.
Don't worry, you got it.
More effective treatments and vaccines. And life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers.
When the company presentation will commence shortly, please enter the live zone and join us. Thank you. Okay, so I hope you enjoy your rest time. So sadly, we only have 3 companies left at the BioLive 2022 BioLive. So without further ado, let me introduce the next company. New Ein, and the speaker will be the director PK Kim. So, PK Kim, please come forward to the stage.
Can I start? All right. Hi, everyone. My name is PK. I'm here to give a brief introduction of our company, Newine, and what we do as a medical device R&D company that utilizes electroceutical technology to treat various chronic diseases. So we started this project in 2017 to develop a wearable medical device that uses electrical stimulation to induce various biological functions that can help with treating various chronic diseases, ranging from dry eye disease, glaucoma, tinnitus, facial palsy, and even cancer. The term electroceuticals is the term originally coined by GSK in 2013. It's an idea that using electrical devices to mimic the endogenous biological signals to instruct the body to regenerate efficiently or function normally again. So we have three themes at the moment with neuromodulation, having the abnormal activity of nerves to function normal again, and tissue regeneration by inducing various proteins and growth factors to be expressed in a way that can help with faster wound healing and even nerve regeneration. And we are also looking at the proliferation inhibition that is in suggestion of interrupting the uncontrolled cell division and ultimately to be applied for cancer therapy. Whew. I just had a heavy lunch, so yeah. So with the technology, we can have a wide range of applications. So as mentioned, we are using both neuromodulation, tissue regeneration, and proliferation inhibition to focus on dry eye disease, corneal pain, glaucoma, and age-related macular degeneration through promotion of corneal and retinal nerve regeneration. We are also looking at the modulation of trigeminal and vagus nerves to treat migraine, tinnitus, and even autism spectrum disorder. That's with the actually inducing there are a lot of the patients with autism and also with ADHD in terms of insomnia. They can't sleep well, so by stimulating our trigeminal nerves and modulating our peripheral nervous system, ultimately the autonomic nervous system, to induce and maintain sleep for 8 hours. So ultimately, we can help with the promotion of neurodevelopment. And we are also looking at the another nerve regeneration aspect for facial nerves. So we are looking at the facial palsy. And then we have, something for cancer as well. So I'll go a little in depth in the later presentation. So for the corneal and retinal nerve regeneration, we have a VR goggle looking like device that the patient wears for 30 minutes a day. That actually helps with the relieving symptoms of dry eye and corneal pain. Also, we are looking at glaucoma and AMD. It's an idea that we are addressing these issues neurologically, and with the exposure to electrical stimulation that forms electric current on the cornea or retina, we have a number of evidences, quite a evidence that suggests the higher expression of growth factors like NGF, HGF, SPRR1 alpha. And all these proteins or growth factors are in fact proposed for treatment for diseases like glaucoma and AMD. So currently, for corneal diseases, we just completed 2 sponsor-led clinical trials in Korea, and we are currently planning for a phase 3 later this year. And for glaucoma and AMD, we are currently conducting phase 2 clinical trials, both in Korea. So hopefully next year, I'll be more than happy to share some results from those studies to you guys. Yeah. We also have a wearable device using a number of modalities, such as electrical stimulation and also giving off the sound to help with the tinnitus. Looks like a earphone. We also have a device for facial nerve regeneration. And yeah, for cancer, so all the indications that I just briefly mentioned about, from dry eye disease, glaucoma, autism, all of them are using the electric stimulation, but they are in the form of electric currents. But we are currently developing a device that emits high frequency electric field pulse generator that delivers specific energy to the region with the cancer. And in fact, we are currently in both cellular and animal studies that that's been suggesting disruptions in cell division process. So we are currently developing a animal model for non-small cell lung cancer, and we'll be along with the development of investigative device for clinical trial sometime next year. So yeah, we are doing a lot of indications for treatment protocols, and these are some of the ideas we have for how to generate revenue, which is very important. This is some our financial forecast of next 5 years for developing our device and putting into the commercialization. And we are also open to collaborative efforts, so for R&D and tech transfer. And these are some of the institutions and organizations that we are working with. We are there's yeah, there yeah, quite a number of them. Yeah, so we started this project in 2017, so it's been 5 years. So there are some of the pictures that we took with things that we've done. So yeah, that's pretty much it. Thank you. And it seems like there are some questions in the audience. I'll be more than happy to answer them one by one. I think we have lots of time left.
Yes,
Yeah.
we have 10 minutes, more than 10 minutes left. So
That's a plenty.
yeah.
All right.
So from the floor, our audience, we have a bunch of time, plenty of time. So if you have any questions or something that you want to add to his presentation or his company, please raise your hand. Any other questions?
In fact, we set up a booth with the Seoul BioHub with some of the devices that we are doing clinical trials with. I'll be more than happy to answer the questions. If you're too shy to say it out loud here, then
Sure.
yeah, you can ask me in person too. So,
Oh, okay.
Oh,
Here's
yeah.
a volunteer there.
Okay. Thanks for the nice talk and your introduction of your companies. I have some short questions. I'd like to know about the mechanism of the treatment for the eye disease using the technology. So I know there are a lot of similar the technology for the neuromodulation. So, in my opinion, so especially regarding the eye disease, so how to proceed to the disease, the mechanism.
Okay. So initially we were mainly focused on the modulating the nerve activity. So with using the electrical stimulation to possibly address the issues with things like depression or schizophrenia, like the psychiatric disorders. But also, while we are developing this particular project, we are working with the clinicians for the stroke rehab. And we had some observed evidence for nerve regeneration. So there were studies that suggested the higher expression of things like BDNF upon the electrical stimulation on the region where the stroke happened, right? So we are thinking that, and then working with the clinicians, they're very conservative. So despite how rigorously we study scientifically, it's very hard to convince, especially claiming that nerves can actually regenerate. So we wanted to translate that and sort of demonstrate that on a more easier ground. So we selected, we chose the eyes essentially. So that was the start. So we are looking into all the corneal diseases and the retinal diseases, but in particular for cornea, it's one of the most sensitive part of our body. It's highly densely innervated, and when we look at the dry eye disease, for instance, previously it's been called the dry eye syndrome, but they just recently addressed it as a disease, and it has a multifactorial reasons why dry eye disease happen, right? But we are addressing both dry eye and corneal pain neurologically. So we wanted to see if because of the nerve damage that dry eye occurs, we wanted to address those in particular. So both for in vitro and in vivo, we set up a animal model on the dry eye rabbits that undergone pretty much laser surgery, like LASIK. And once we did that, we applied the electrical stimulation that forms electric current only on the surface of the eye, so on the cornea, and we observed the before and after, and also the ones that were stimulated and ones that didn't, that weren't receiving the stimulation, to see if the nerves, whether or not the nerves regenerate. So we did the study, and we saw the physically the nerves being regenerated. I'll be more than happy to show you that results in our booth. But for the time being, we saw those. We also saw the. It's also important that if the nerves are generated, it should function normally again, right? So we looked at its corneal sensitivity, and at the same time, we are looking at how fast the wounds were healing. So we are looking at the corneal staining and the conjunctival staining scores. And then we are also looking at the mechanisms of, it's still a bit of a black box, but we definitely saw a higher expression of both the NGF, EGF, BDNF, and SPRR1 alpha. And all of them are actually some of the proposed options for treatments of dry eye disease, particularly with the neuropathic causes. But interestingly, what we've been sort of scratching our heads with is there's been the results for the lipid layer thickness. That's actually something to do with the secretion of lipids from the meibomian glands, and we don't think it's because of the promotion of nerve regeneration or anything like that, but probably we are also stimulating our trigeminal nerves and honored with the autonomic nervous system. So there's some mode of actions that we think are working in parallel to achieve those indications of faster wound healing and actual nerve regeneration. And from our clinical trial with the dry eye disease patients that undergone a laser surgery, that's what we are working on. The patients were given with the device on the day of the surgery to take home and wear the device for 30 minutes a day. And after 2 weeks and for the 12 weeks of monitoring, we saw definite improvements in comparison to the ones that were receiving the traditional post-surgical measures. So now we are sort of going a little further with it and wanted to see the promotion of hopefully the nerve regeneration in the patients with the AMD and glaucoma. Yeah.
Thank you.
Thank you.
Well, thank you very much for the insightful question and answer. Any other questions? If there are no more questions, that it has been UI. Thank you very much. Thank you very much. Yes, as he mentioned, he has a booth in the exhibition hall. So if you have any question or curiosity, you can visit his booth. Thank you very much. Coming up next company, Health Biome, will start at 2:30. So I'll see you in 10 minutes. I'll be back. Thank you.
Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines and life-changing breakthroughs. And we never stop in pursuit of answers.
네, 바이오 라이브 이제
So now we are going to begin the presentation. Now we have only 2 presentations to go. So you are looking around this exhibition hall. So if time allows, please do come in the Bio Live Zone and attend this meeting.
please enter the live zone and be seated. Thank you very much for your attention. Okay, welcome back from your break. That was just short break. Well, as I mentioned earlier, we only have 2 more companies left to be presented in BioLife 2022. Okay, quickly introducing the next company is Health Biome, and I would like to introduce the CEO of Health Biome, Mr. Kim Byung-chan, the CEO of Health Biome. Come to the stage, please. Come forward to the stage.
예, 저 방금 소개.
Hello, ladies and gentlemen. I'm CEO Kim Byung-chan. As introduced, I'm from Health Biome. So let me begin my presentation. So I'm here in the Bio Korea. While taking this opportunity, I'd like to introduce my company because we are the newly established company. As you see here, we are focused on the microbiome products. So one thing I'd like to emphasize is our microbiome is different from other the available microbiome. Lactobacillus is different from the strict microbiome that is developed by my company. So I'd like to share why we focus on strict microbiome in the later part of my presentation. This is the agenda of my presentation. I'm going to introduce my company, and after I'm going to share the merit and advantage of a strict microbiome, and that will be followed by competitive advantages of my company. So the microbiome is located in Daejeon City. This is the research arm which receive a research grant from the government. So we have the office, which is about 40 pyong in Daejeon City, and also there is another the office area in nearby Daejeon City, and in Seoul we have another office, and we have about 15 employees in 3 different locations. I used to be research fellow of KRIBB, and also I have the head as the researcher in KRIBB. In November 2017, I established this company based on the technologies I developed in my years in KRIBB. We mainly they receive the grant from government in order to develop the innovative technologies, and we completed the Series B in the year 2021. I'd like to introduce myself first. For more than 20 years, I have been studying the microbiome. So my specialty is a strict microbiome. I isolate and purify the strict microbiome. That is my specialty area. Based on this technology, I do the genetic and the manipulations, and also produce the high value added products based on that. I have more than 20 years of experience in this field. During my years in KRIBB, I understood that this strict microbiome can give great benefit to human health. So I secured this microbiome resources, and I isolated about 3,000 these strains, and I have about 3,000 the target microbiome. And out of that, I developed the products by further developing 100 microbiome strain. So, Mr. Yang, Mr. Lee, and these the other research fellows who are developing these technologies together. The strict microbiome has a great benefit, so let me explain that first. So those who are engaged in microbiome business know about the merits and advantages of microbiome. Microbiome has the great the role to play regarding human health. So when we talk about the microbiomes, that include the proteins and the metabolome, and as you can see here, the microbiome has a close relations with other disease, the brain disease and also intestine disease, and those have the close relations to the intestine microbiome. So in the United States, there is an open biome. So their main business is offering the feces to clinics and hospitals. So this is the information about the feces transplantations. And there are a lot of cases, but because of time constraints, I'm going to talk about only the cancer and dementia. You can see 2 papers at the last 2 years ago. This paper was published in the United States. And this case is the Alzheimer, the patient, 82 years old. The fecal microbe transplantation was done for this patient. So, well, these patients had a 29 the grade out of 30. The 29 means that this dementia patient had the recovery of this the illness. So this is a quite the viable the evidence. This paper is from the Nature. So PB1 the anti-Zen and the trial was done. The subject was the enrolled and the fecal microbe transplantation was made for this 16 subject. And the phase 2 clinical trial was made. So I introduced the case of oncology and the dementia, which are quite the problematic disease we face today. So what I want to say is, in both cases, these feces microbe have a great benefits. Then, what is the composition of this microbe? So not many people knows about this. Actually, in the healthy adult, there is no the lactic acid. So NGS analysis is done, so we could not see any of that. And in the year the 2019 in Science, the journal, the information was released. So actually, key player is not a lactobacillus. The strict microbiome plays a critical role in human health. So let me take a few examples here. So when you see the fecal, the microbe, there is a blockbuster strain. So that the newly emerging star is Acamedia mucilus. So, actually, the strain we are developing is this strain, which is receiving great attention. For the first time in the world, for this strain, we have the patent. So this strain has the great benefit regarding degenerative disease like the dementia. So we are applying additional the patent for this strain. This is an annual the cognitive test. So this is the healthy mouse, and this mouse had water, and this mouse had the lactobacillus. So there are 3 different groups. And this study was done in Europe, and the result was released last year. So this is the test done and with the mouse subject. So, by giving the fecal microbe, the mouse had the shorter time in finding ways in maze. So the mouse with water, the mouse without lactobacillus did not show big differences in the period of time that took in order to find the pathway in maze. But the mouse which had the fecal microbe had a much shorter time in finding the pathway in maze. So this is the additional data. So the cancer, the growth is visible here. With the lactobacillus, there is no big differences, but when this the particular fecal microbe, the train is given, and you can see the differences. So the next generation micro biome was studied, and the related data was released as you see here. So all these the data is about strict microbiome, and you can see the plus and minus signs. The plus is the benefits. So when it comes to the strict microbiome, and it has a great benefits regarding cardiometabolic disorders and inflammation conditions. But it is difficult to culture this microbiome, and there is no the safety data, and also there are other challenges we face regarding strict microbiome. But still, based on the paper, we could understand that this microbiome from feces has a great benefit indeed. One is the acabromyces, and another one is the strict microbiome. And well, this is the newly coined name because I found this one and also isolated this, and I had the patent for this one as well. So the second strain is the in-house strain that is developed and isolated by ourselves. You can see the facilities to ferment this strain, and also the products can be produced by using these facilities. So culture and also safety analysis are done here, and 500 milliliter products were produced. IRD approval was made, and currently we are doing the clinical studies. And we should have the additional clinical trials in order to in order to be recognized as a treatments. And now we are doing clinical trials in order to be recognized as a functional food. So in order to be recognized as the meaningful treatments, additional clinical trials should be made, and they should be done in the future. So Akamentia is a great strain, but the one which is the BH502, that is the another strong strain. I myself found this one and isolated this strain. This is additional information. This is the Alzheimer mouse test. So after receiving these fecal microbe injections, the level of the cognition has been increased. And this is the brain functions. And when we conducted the animal test, we could not see the recovery to the normal status when the lactobacillus was injected, but the microbe is injected, then recovery was made to the normal stage. In the year 2024, we are going to have the additional clinical study. So next, I'd like to share information about oncologists. Well, before we inject the tumor, the tissues, and this fecal microbe is given to the mouse, and the treatment is done for the next few weeks. Rather than in vitro test, we have a great specialties in animal test. During our years in KRIBB, we conducted number of animal tests. That's why we have a great specialties. This is another strain we developed by ourselves, and this have been transplanted in the skin, the areas, and this shows great benefits in the cancers, and in particular, and we have great the benefits when it comes to pancreas, the cancer. So the clinical trials we did is a monotherapy that was not concurrent therapy. So when monotherapy is designed in these clinical trials, we could see great benefits. So as I mentioned earlier, National Cancer Institute and had report regarding the mechanism. The information was released in October last year. So you can see the profound informations. So actually there have been recognition regarding the benefit regarding oncologists. There are many different the oncology-related indications, and fecal microbe has a benefits in the quite extensive indications in oncology area. Dementia has had a combination with a psychedelic MB. So, well, in short, that is called the TMB. So this is the mechanism. The strain, the protein can be activated in this case. So stimulation, the interferon can be induced in this process. So NK cell is activated in this process. So the NK cell is increased by 3 times and infiltration is done. So interferon level is increased, and the elements as you can see here is increasing as well. So there are the macrophage which promote oncology, and there are also other things that the reduce and this oncology the activities. So, well, based on this analysis, we could develop other products to cure the oncologists. The SDP will be available this year, so the production will be done. The toxicity data will be secured at the end of this year. So by the end of next year, we are going to make submissions for the clinical trials. So the clinical site will be both Korea and the United States. And the next I'd like to talk about the functional, the food elements. The meta-analysis is done, so based on this analysis data, I could say that fecal microbe have a great benefits. And the analysis regarding the analysis, my company has a great the strengths indeed. So we collected the breast milk and the analysis. As I said, we have the greatest strengths in order to cure the degenerative disease. So last October, our study result was shared in the medical journal. So in the aging process, well, one thing that is visible is the muscular strength is the reduced, but the total the weight of muscles and the level of muscular strength have been increased after applying these difficult microbes. So that is another evidence, and this is another the example. And we had the BHB05 the analysis. Inflammation was induced by using these the materials. As time goes, the concentration can be changed, and that concentration will be changed from low level and medium level. So HB05 give the benefits when that is given. So Professor Zhang in KAIST and our research team worked together in order to conduct this study. And we had a human body the tests already, and the result will be available next year. So like this, I introduce my company and available technologies developed by the Health Biome. If you have any questions, I'm going to accommodate that now.
Do you have any questions about his presentation? Okay. So if you don't have any questions, there are no more questions from the floor, then this is the end of his presentation. Thank you very much. It has been Health Biome. So we only have one last presentation left, but as we promised you, the last presentation will be starting at 3 o'clock Korean Standard Time. So we're going to take a short break. I'll see you soon at 3. Thank you.
every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got it.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got it.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. Every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got it.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers. every single day for the good of millions all over the world. We rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers.
네, 바이오 라이브 마지막 발표가
Now we're going to start the last presentation for BioLive. Please come to the BioLive zone.
Please, the last presentation of BioLive 2022 will be starting in 1 minute. So it would be appreciated if you all come into the live zone and have your seats and look forward to your attention. Thank you. Okay, ladies and gentlemen, we have finally reached the last presentation of BioLive 2022. And CEO Hyun-Soo Park from Cepho Bio will do the honor of ending the session. So, ladies and gentlemen, please join me in welcoming her with a big round of applause.
Thank you. So we are exploring the next generation cell therapy for regeneration. In the aging society, musculoskeletal disease patients are gradually increasing more and more. Up until now, biomaterials are available, such as hydroxyapatite and calcium phosphate, but they are simple substitutes and not perfectly designed to cure disease in the long term. Cephobio has currently introduced allogenic pre-osteoblasts, namely pan-cell bond to cure bone fracture and osteonecrosis. Bone regenerations occurs by a series of events such as osteoinduction, osteoconduction, osteogenesis, and angiogenesis. So pan-cell bone is pre-osteoblast, which is differentiated from the umbilical cord stroma MSC. They mimic osteogenesis pathway of bone marrow stem cells. We identify their characteristics such as surface markers, transcription factors, and secreting proteins. Those characteristics match their mode of actions. Pan-cell bone has dual functions. One is bone formation, and the other is paracrine effects. The former is relevant to osteogenesis and osteoinduction, while the latter is relevant to osteoconduction and angiogenesis. We use those characteristics as potency markers to expect their action. Let me explain how we differentiate stem cells into bone cells. History goes to 2012 when we collaborated with British chemist Professor Rain Yulin to investigate whether hydrogel could provide biomimetic culture environment by modulating their stiffness. Luckily, we found the optimal range of stiffness of hydrogels for osteogenic differentiation. As you can see, the first raw is alizarin red staining of osteogenic differentiation of various stem cells by conventional 2D culture dishes with cytokines. Red color identifying calcium-containing osteocytes. The indicator of this is the indicator of bone cell differentiation. At 2 weeks, duration after the differentiation, induction alizarin red positive cells are 100% in dental stem cells, but hardly found in umbilical cord MSC. Depending on tissue origin, result varied and takes ages of long time for differentiation. However, when hydrogel was applied in 3D culture system, which is on the second row, alizarin red stained cells are found within 5 days, irrespective of tissue origin. This technology is patents registered in Korea, Japan, China, United States, and Europe. So, and our first application is avascular necrosis of the femoral head, which is caused by the interruption of blood supply at the head of femur and osteogenesis. There is no treatment except total hip replacement in the final stage. We are preparing to register pan-cell bond as an orphan drug for this rare disease. We extend indications to the more common diseases such as osteoporotic fracture and spinal fusion with the same mode of action. Our next pipeline is cancer immunotherapy using directly induced NK cells. This technology was developed by Dr. Yeok Cho of CREEP, licensed in 2020. Recent cancer treatment is moving towards cancer immunotherapy. Natural killer cells have been paid attention since the serious adverse effects of CAR T cell therapy were reported. This diagram shows how to induce NK cells from PBMC. PBMCs are exposed to Yamanaka factors in Sendai virus for 4 to 16 hours and cultured in specific differentiation medium. As you may know, the Sendai virus is not integrated into chromosomes. Within 2 weeks of time, over 80% of cells are differentiated into NK cells, and through another 25 days of culture cells are enriched to nearly 100% NK cells and expanded over 1,000 times. This process is simple. Comparing to our competitor, Fate Therapeutics INK cell, which is composed of 3 steps: IPS induction, HSC differentiation, and finally, NK cell differentiation. In addition to simple protocol, the uniqueness of our direct NK cells is their dual functions: CD56 bright / CD16 positive. Normally, NK cells have either phenotype. Therefore, NK cells highly produce interferon gamma, which is phenotype of CD56 bright cells, and have killing activity, which is the phenotype of CD16 positive cells. So their powerful killing activity was further confirmed using mouse xenograft model. This process of cancer stem cells has been reported to remain after the surgery and spread to invade other organs. Thus, CD133 positive cells, cancer stem cell markers, are selected to form cancer spheroid. When NK cells are cultured with this spheroid, NK cells penetrate into spheroid and kill them. These unique NK cells behave like memory, like NK cells, and recruit T cells. This slide shows the pipeline. AVN is in phase 1 clinical trial and is expected to complete trial by June. In phase 1 clinical trials, exploratory efficacy is analyzed at 3 months after cell transplantation by MRI to measure bone regeneration, VAS scoring to measure the degree of pain, and WOMAC scoring measuring bone function, and Harris Hip scoring evaluating the quality of life. And so far, we found promising results, although the number of patients are limited. Other indications are followed as shown here. And we have our own GMP facility with 3 manufactories and certified as advanced biomedicine manufacturer in 2021 by Korean FDA. Finally, I would like to emphasize our naive stem cells produced by simple cultivation and failed to prove mode of action. Further, the first generation of therapy is limited and unfulfilled for the following 3 reasons. One is low or unpredictable rate of production, and the second is intractable quality control, and lastly, low cost effectiveness. Cepho has recently succeeded in mass production and quality control of allogenic cells to create master cell banks. Next, Cepho's differentiation technology allowed master cells to transform into functional therapeutic cells. Finally, our cryogenic technology helps us to supply those functional cells in a frozen status in order to overcome shelf life, transportation, production capacity, and quality control. This is the end of our slide. Thank you for listening to our presentation. Thank you.
Excellent presentations. So as I told you, his presentation was the last one in BioLive 2022. So okay, so we'll take some questions from the floor. Do you have any questions or comments? Any? Okay, so you can also send a question to the presenter online, so you can scan the QR code or access BioLive on the homepage. So if you have any question, just feel free to visit our website as well. So if there are no more questions here, so this is end of Cepho Bio. Thank you very much for your presentation.
Thank you.
Thank you. Okay. Well, all presentations of BioKorea 2022 BioLive have ended. We are truly grateful to all the attendees and presenters who have joined this event. And we hope that BioKorea 2022 BioLive became a good opportunity to strengthen business and networking through active exchanges between participating companies. Then I would like to announce the ending of BioKorea 2022 BioLive. Thank you very much once again. I look forward to seeing you next year as well. It has been Kim Kyung-mi. Thank you very much. See you next year.
Every single day, for the good of millions all over the world, we rise and get to work so we can help bring you better tests.
Don't worry, you got this.
More effective treatments and vaccines, and life-changing breakthroughs. And we never stop in pursuit of answers.